I want to thank everybody for coming to this Monday afternoon session. This is the session on divergence, coexistence, and hybridization. My name is Becky Fuller. The question wrangler is Bill Resiteritz, and we have our tech person, Mark McPeak. The first talk given this session is by Andreas Härer. The title is Non-Parallelism of the Gut Microbiota During Parallel Evolution of Vertebrate Hosts. I'm going to share my screen here. And make sure you get this. I'm going to hit play on this. Sorry, let me take this off here.
Hi, everyone. My name is Andy. I'm a postdoc at the University of California in San Diego, working with Diana Rennison. And today I want to talk about parallelism of gut microbiota changes during the parallel evolution of their hosts. And in particular, I want to focus on what happens to the gut microbiota when their hosts adapt to novel trophic niches and in cases where this happens in parallel. We know that trophic adaptation can be a driver of ecological divergence and subsequently also lead to speciation. And there are famous examples like the cichlid fishes from the Great Lakes in East Africa, but also the Darwin's finches on the Galapagos Islands. And in both cases, we do see a spectacular diversity in terms of morphology, but also ecology. And this diversity evolved in part due to adaptation to different diets. We also know that diet can affect the composition of gut microbial communities. And this has been shown in multiple species and also in well-studied model systems like mice. And taking together this information, we can ask whether we can actually predict changes of the gut microbiota based on our knowledge on the ecological divergence of their hosts. And with ecological divergence, I'm specifically referring to divergence in trophic ecology, so in diet. And this question of predictability can best be addressed in systems where evolution repeated itself or in cases of parallel evolution. Parallel evolution is a widespread phenomenon in nature, and it is regarded as evidence for natural selection, since we wouldn't expect the repeated and independent evolution of similar phenotypes just due to random processes. And however, we have to be careful when we study parallel evolution because the extent of parallelism can actually vary considerably across independent replicates within study systems. Here's one example of a study on three-spine stickleback. And here the authors investigated lake and stream population pairs. And what they found is that across replicates, there's a lot of variation in the extent of parallelism. And here they looked at different morphological traits. Another theoretical study by Thompson et al. Found that the predicted genetic parallelism is actually stronger when the selective landscape is also more parallel. But there's a lot of variation depending on the selective landscape. And by combining the results of those studies, we can actually see that there's a lot of variation in parallelism, and it's not just a binary phenomenon where you would score something as parallel or non-parallel. And the question of how parallel are evolutionary changes or how predictable actually is evolutionary change has been debated for decades, starting with Stephen Jay Gould, who proposed the thought experiment of replaying the tape of life and whether we would see similar outcomes across replicates. And it's also debated today. And there was a book published by Jonathan Losas a couple of years ago, still dealing with this topic. And there has been another study by Dan Bolnick et al. Published 2 years ago, where they actually argue that we should consider parallel evolution as a quantitative continuum rather than as a binary phenomenon. So, and they also proposed that by considering information about this, where actually populations are on this continuum, we can actually find factors that affect the extent of parallelism. And they proposed one method, which is a multivariate vector analysis. to actually study and quantify the extent of parallelism. And what this analysis does is actually, so what you can do is, let's say we have two different ecotypes of a species, ecotype A and B, and then we do have the similar ecotypes across different environments, in this case, two different lakes. So what we can do is we can measure some traits, maybe morphological or genetic, and then we can conduct a vector analysis and to connect the means of ecotype A and ecotype B. And then we can quantify the extent, but also the direction of change. And then we can compare the direction of change across independent population pairs and see how parallel they are. So in this case, they change in the same direction. So this is an angle of around 0 degrees, and this would be highly parallel. There could also be cases where we don't see parallelism, so the direction of change will be quite different. In this case, 90 degrees, which we would see as a null expectation for non-parallelism. And using this analysis, we can actually address the question of how commonly do we see parallelism in gut microbiota changes when their hosts evolve in parallel. And we applied this method to different systems of fish. And fish are actually a good group to study parallel evolution, because there are many cases where populations independently adapted to similar trophic niches and representing cases of parallel adaptive evolution. And for many of those systems shown here on this slide, we also have information about their gut microbiota. So what we did is we reanalyzed those published data sets and to ask whether we do see variation in the extent of parallelism across those study systems to see if we can find some general patterns. So what we did is we asked this question using two different approaches. The first would be to compare population pairs. So here's an example of cichlid fishes from the country of Nicaragua. And in two small and young Greater Lakes, Greater Lake Chiloa and Greater Lake Apoyo, benthic and limnetic species evolved. And this happened repeatedly in those two Greater Lakes. So what we can do is we can actually compare the direction of change between benthic and limnetic species across those two lakes. What we could also do is using an outgroup and compare the extent of parallelism from this outgroup compared to different focal populations. So for example, sticking with this example of cichlids, those Greater Lakes were actually colonized from Great Lakes. And so we can infer that the fish from the Great Lakes are the ancestral state, and we can see what happened after fish colonized the Greater Lakes and how much parallelism we can find in terms of their gut microbiota. So coming to our results, here you can see different studies from different fish systems, and on the y-axis you will see the angle between population pair comparisons for those different systems. And what you can see is that there is actually quite some variation within, but also across systems. So for example, here in the first column, you see cichlid fish from Lake Tanganyika. And you can see that for some comparisons, angles are below 40 degrees, whereas for others, it's close to 90 degrees. So again, so that means that there's a lot of variation. And remember that all of those study systems are actually examples for parallel evolution associated with divergence in trophic ecology. And what we also tested is whether we actually do see significant parallelism, and we measured this as by testing whether the angles are lower than 90 degrees, which was our null expectation. And you see that for two systems, the Lake Tanganyika cichlids, but also for benthic and limnetic sticklebacks, there's actually evidence for significant parallelism. So this is based on the taxonomic composition of the gut microbiota that we determined based on 16S sequencing. And based on this data, you can actually infer the functional microbiome. So you can infer information about metabolic pathways present in the microbial community. And we did the same analysis And again, what we find is a lot of variation within and across study systems. And here in this case, only for the Lake Tanganyika cichlids, we do find evidence for significant parallelism. So there's a lot of variation. So what we asked next is whether the angles for the taxonomic composition, but also for the function of the microbiota, are correlated. And as you can see here, yes, they are strongly correlated. So based on this, we now wanted to know whether angles are smaller for either composition or function. And what we found is some suggestive evidence that angles are actually smaller for the functional microbiome, but it's not statistically significant. But then we only focus on a subset of our data, and this is a subset that shows some evidence for parallelism, and that means that the data has angles smaller than 90 degrees, both for the taxonomic composition and for the function of the microbiome. When we only focus on this subset, we do find highly significant differences between function and composition. So the angles for function are much smaller. That means that parallelism is stronger for the functional microbiome. And so the question is, why do we see those differences? And it could be that due to functional redundancy across bacterial taxa, we do see more convergence on a functional level, since the taxonomic composition might be affected by the environment, for example, but different bacterial taxa might converge in the same functions. And this might be indicative of adaptive changes of the microbiota. Then we did the same analysis for the comparison of outgroups versus focal species. We did this for 3 systems, and both for the taxonomic composition and for the function of the microbiome, we do find evidence for parallelism. But interestingly here, for the cichlids from Lake Tanganyika, they actually have the largest angle, so they're the least parallel compared to the other systems. And we think that this could be explained by the fact that those fish in Lake Tanganyika, the outgroup and the focal species, occur in the same environment, so the same lake. Whereas in the other 2 examples, in sticklebacks and in the cichlids from Nicaragua, the outgroup occurs in a different environment. So this parallelism might be driven by, in part at least, by environmental differences between the outgroup and the focal populations. And again, we looked at the correlation between angles for composition and function, and they are strongly correlated, but we did not find differences between the angles for function composition. And this also holds when you look at this subset of data points that show evidence for parallelism. So that means, again, having angles of lower than 90 degrees for both measures. So there seems to be a different pattern here compared to the population pair comparisons. And with this, I hope I could show you that this multivariate vector analysis provides an interesting tool to determine levels of gut microbiota parallelism across different host organisms. And as you have seen, our analysis so far has only been restricted to a few examples of fish. And what we are currently working on is extending this to a broader taxonomic and phylogenetic diversity. And luckily nowadays we have the gut microbiota characterized for hundreds of different vertebrate species, and we have information about their diet and about their divergence times and about the environment where they occur. And now we want to know if we can find more general patterns and factors that actually explain the extent of parallelism across a broad diversity of host organisms. And with this, I would like to come to my conclusions. So parallel evolution of the host does not necessarily translate to parallel changes of the gut microbiota. And we found this in different systems and different examples of parallel evolution in fish. We found some evidence that parallelism actually might be stronger for the functional gut microbiome compared to the taxonomic composition. As I mentioned before, this might be due to functional redundancy across bacterial taxa. And I hope I could convince you that multivariate vector analysis is a promising tool to study the factors that actually promote or hinder gut microbiota parallelism, and that we can use this method to infer general patterns and learn more about gut microbiota dynamics in nature. With this, I would like to thank all my collaborators that worked with me on different projects concerned with the gut microbiota, and in particularly, I want to thank Diana Rennison, who worked with me on the study I presented today. And of course, I would like to thank you for listening to my talk. And yeah, thank you very much.
Cool. Thank you very much. I think we have time for questions.
Remember, you can send them in on Slack or the chat function.
Maybe before the questions come, I quickly want to mention, I realized I forgot to thank the organizers, of course. It's been such a wonderful experience so far. Thank you.
So, oh, here we go. Nick has a question. Thanks for that talk. You lose some information by using just the angles. Have you looked at what information can be gleaned for the magnitude of the change?
Yes, that's actually, that's a good question. And this is something that we've also done. So I left this part out for this talk today. But yeah, we can also quantify the magnitude of change and compare like whether if there's a stronger magnitude in change, how this correlates to the parallelism, so regarding the angles. So what we found is that that the stronger the stronger the change, so the higher the extent of change, the more parallelism we see for the population pair. So that means if you have a lot of change, there's a higher chance that this is actually in parallel. So it might be that those stronger changes might be indicative of adaptive changes. Yeah, but there is some correlations that we find between the extent and the direction.
Blake has a question. How much do we know about the distribution of heritability for microbiomes of fishes?
Yeah, that's a sorry, that's a very interesting question. I don't think there's much known about this, and this is actually something we are also interested in, and we're currently working on developing ideas on how to test this in some of our model systems, so in sticklebacks. But I think outside of other model organisms like mice, I think there's not much known about the heritability of the microbiome. And I think I would assume this could vary quite substantially, depending on reproductive strategies, on parental care, and so on. So I would think this varies quite a bit across study systems.
All right.
Bill,
don't see anything just now.
Can I ask
Becky?
Yeah, sure. Yeah, so I mean, the heritability question sort of ties into the question I had too. So, I mean, they presumably pick some of this up, right? When they're out eating stuff, right? Yeah. So, I guess are those microbes in the water, or are they on the actual food items? Like, do we know like where they're picking those up and how much? Like it just might be that you might like some microbes. I don't know. Are the same microbes everywhere, or yeah? I guess I was wondering about chance events of picking stuff up.
Yes, that's a very good question. And I mean, just generally, it's the microbiome. It's a very messy thing to study, right? This is highly dynamic. It can actually change within days once you change diet. So it's highly variable over time. There have been some studies where they looked at the contribution of what are the sources of microbes. And I think, if I remember correctly, in this one study, there was some indication that actually the microbes associated with diet, like on the prey items, are actually predicting the changes or affecting the microbiome of the predator. But yeah, I mean, this is totally variable, and like when fish occur in different environments, like let's talk about fish. I mean, different lakes, there would be different microbial communities in the water, which would lead to more stochastic changes in the microbiota. And this is where I think it's interesting to compare the function and the composition of the microbiota, because you might expect less stochasticity related to the function. But again, this is not a perfect method to infer the function, but yeah, all sorts of sources.
Yep. Cool. Thank you very
There's
much.
one more question that
Thank
you
you.
could get to on the chat later.
Okay. Yeah, I'll do that. Thank you.
Our next speaker is Nicholas Kortessis. The title is Character Displacement in the Presence of Multiple Trait Differences, Evolution of the Storage Effect in Germination and Growth.
Great. Thank you very much, Becky. Can everybody see this okay? Good. So yes, first of all, thanks for coming to my talk, and for everyone who organized this. It's nice to be able to have some interactions with scientists from all over the place, even though we can't be in a CLMR together. All right, so I'm going to begin by talking about one of the big questions in ecology and evolution, which is about species diversity. In community ecology, we tend to focus on species coexistence. And here's just one example of why this is just a big question in the field. So if you go out to southeastern Arizona, so here's a picture from spring 2019, just outside Portal, Arizona. This is a relatively famous place in ecology. The Chiricahua Mountains are here in the background. It's a great birding spot if you want to go there sometime. If you go in the winter, following a rain, a diverse community of annual plants can be found there. So in approximately 50 hectares, so think tropical forest plots, you can find over 50 species of these annual plants that all show up in the same 3 or 4 months together. And you're just struck by this question of what allows for all these species to be maintained in the face of interspecific interactions. So in this picture, you can see that they grow in quite dense clumps. They're all doing effectively the same kind of thing. They need light, they need water, typical community ecology problem. So what allows them to coexist if they are doing that at all? Well, we know that any answer to this question requires that the species differ some way in their ecology. So here's just a subset of some of my favorite species that you can find out there. And you can just see from looking at them, you don't have to be much of a biologist to figure out that they're pretty different. And part of the task of answering how they stably coexist is figuring out which of these differences matter and which do not. But there's another question that's evolutionary in nature, which is how do these differences arise and do they persist when species come into new collections of in communities? So when they assemble and they come into contact with new species, do those differences between them get larger, or do they erode over time? So this is essentially a graphical depiction of what I'm talking about. Here we've got evolutionary time on the x-axis, some trait value that's ecologically important and determines competition on the y-axis, and their position here tells you how similar they are, but I've also made it by color just to help out. So here we have adaptive divergence of 2 species that compete, and whatever the coexistence mechanism that results from this adaptive evolution is pretty robust. We expect whatever the differences are between the species with this particular mechanism to get larger. An alternative is that we could observe some species with some differences as they assemble in a community. And then with sufficient time, those differences may erode, leading to the ultimate collapse of coexistence of those species. For in this example, the one that eventually turns purple goes extinct. So these kinds of models of character change are just reflections of mathematical models of character, the process of character displacement. So I'm going to talk about character displacement not as the kind of sympatric allopatric comparison pattern that was the origin of the term. I'm going to talk about it as the evolutionary process that reflects adaptation in the presence of competitors. So the theory about character displacement is really dominated by MacArthur-style Lotka-Volterra models. And if you're not familiar with them, I'd be surprised if you weren't, but I'm going to give you a quick overview. They look a little something like this. We've got a resource spectrum here. And for 2 species, a blue species and an orange species, there's an evolving trait that reflects a niche position. So that's given by these dashed lines. And the shapes of these resource use curves tell you how much of any particular resource one of these species consumes. And if these curves diverge from one another, we call that character displacement. If they come towards one another, then that means that they get more similar to one another. That's adaptive convergence. And if resource partitioning allows these 2 species to coexist, maybe they'll collapse. So I want to take you back to the annual plants and ask, can we apply this theory to annual plants in the desert? We just have to make a few changes in our mind. The first is that the mechanism of coexistence for annual plants in arid environments is not presumed to happen by resource partitioning. There's a great deal of theoretical and empirical work that points to the storage effect as the dominant contributor to plant species coexistence there. So here's just a simulation I ran of 5 different species that are stably coexisting via the storage effect. And for those of you who are not familiar with it, the storage effect works like this. It's a temporal analog of resource partitioning, but it operates in a fluctuating environment. So the idea here is instead of consuming fundamentally different kinds of resources, they're consuming the same resource, but species partition up their use of consuming that resource in time. Whereas sometimes it's one species takes a lot of their resource, and then at another time, a different species uses up a lot of the resource. So it's time-based partitioning instead of resource-based partitioning. And the ecological differences that are important for this to work are that there have to be different patterns of activity among the species to capture resources. And this is commonly called species-specific responses to the environment. That's what you'll see in the literature commonly. Now, there's another critical component to the storage effect here, which is the populations have to be buffered against poor conditions. For example, when the environment is poor and competition is intense. So when you can't acquire very many resources and there are few to go around in the first place. And I'll get into that in just a moment. The storage effect, I also want to make a note saying that it's not, I'm going to talk about it in terms of annual plants, but it's not specific to annual plants. It's been hypothesized for desert annuals, reef fishes, phytoplankton, perennial grasses, tropical forest trees, among many others. Okay, so here's what it looks like in annual plants in terms of those necessary ecological differences. One of them is trade-offs in germination responses to temperature at the time of rainfall. So here's some growth chamber experiment data where we've got species aligned here by different colors. And then there are different temperatures that we leave them at and we check to see, how well do they germinate. And you can see that the species that germinates best in cold temperatures germinates worse at warm temperatures. And the one that's best at germinating in warm temperatures is worse in cold temperatures. So as the temperature fluctuates when it rains, we get different patterns of dominance of species that germinate and then grow in different years, with the year time being here on the x-axis and different species here being on the y-axis. Okay, so that's one. But there's another one. We also know there's good empirical and theoretical evidence that trade-offs among species in their ability to grow with abundant rain and when rain is when moisture is limiting turns out to be really important for the storage effect too. So there's this paper from 2009 in PNAS that essentially shows there's this trade-off between drought tolerance and relative growth rate of plants. And that in a fluctuating environment can lead to a storage effect as well. So we've got a problem. The theory of character displacement assumes there's one trait. It's this niche position. But we've got a multivariate trait problem. We've got germination responses and we've got plant growth responses following germination. Making matters worse, these two traits don't act independently of one another. They're interactive in their effects on fitness in multiple ways. The first is that plant growth is conditional on successful germination. If a plant doesn't germinate, it can't grow, right? So there's sequential life history activity here that's involved. The second is that germinating in a year of poor growth should be disfavored. So a lot of these environmental conditions are correlated with one another, and there's a whole other body of life history theory in annual plants that suggest that species should germinate most in years when they grow best. And so these two may be coupled together in that way. The final is that germinating a year where plants can grow quite large increases demand for resources. So if germination fraction's high, lots of plants show up, and if it's good for them to grow, they all get really big, and that's intense competition for whatever soil resources are available or light resources. And we know that these interactions can be important because it looks like they've shaped trait correlations in these communities, where we can see that some measure of germination temperature preference is correlated with drought tolerance. Okay, so for my work, I wanted to see how these two traits may change the picture of adaptive evolution in response to competition. So this is a question about whether character displacement changes when another trait also contributes to competition. And to show you that this also isn't just an annual plant thing, here's some data from a study published in 2018 about monkey flowers with a bunch of different species pairs measuring allopatric and sympatric differences between species. And you can see that it's done over 1, 2, 3, 4, 5, 6 different ecological variables. So this is a multivariate trait problem. The Darwin's Finch's beak shape problem is also implicitly a multivariate trait problem. In fact, they use ordination analysis to make the problem tractable. And then here in a more recent study about green anoles, they look at 2 characters, toe pad area and lamella number. So no, I'm not arguing that these papers are flawed in any way, just that multiple traits are often involved in competitive interactions, but our theory doesn't account for it. Okay, so here's the plan. The plan is to have a genetically variable trait, which is our germination strategy. And we'll have another trait that's involved for the plants that's fixed. And this will be a fixed plant growth strategy. And we'll look at how the differences between species in their fixed traits affect selection for divergence or for convergence of their germination strategies. And the model has to be sufficiently flexible to allow for correlations between these traits. So there's some math that goes into this, but I'm just going to show you how it works. There's a trait theta G. That's a germination strategy for species J. If this theta G for species 1 and species 2 are very similar, so if they're have essentially the same value, then if we look at some measure of their germination fraction over time, they'll have very similar responses. So these 2 species are ecologically very similar in their germination traits. We can make it a little bit different, and then you can see that here they're largely independent. And if we make it very different, the maximum difference in this particular model, then they're completely uncorrelated in their germination fractions over time. So this is the maximum ecological differences they can have with respect to germination rates over time. And we can summarize this by looking at the absolute difference of these thetas. And it runs from 0 to pi for reasons I won't get into now. But the nice thing is that it takes us from between species correlation and germination from 1 all the way to -1. And the further we go down this way, the stronger the storage effect is. Okay, so we've got it set up here. We've got differences between species and their germination traits, which can evolve. Species differences in their growth strategies, which are fixed, and then potential correlations between them. And this is what the model looks like. It's an annual plant model. This is the fitness of a seed of species J. It's made up of seed survival and reproduction conditional on germination. And this is where those traits show up. The germination strategy shows up in the fitness of the species and also in competition. The plant growth strategy shows up here in reproduction and in competition. And then the traits for the competitor also show up in competition. So they're all involved in fitness here. I'm going to use evolutionary, I'm going to model the evolutionary dynamics of the system using adaptive dynamics. And the analysis is really going to be done kind of by comparing evolutionary endpoints in sympatry and allopatry. So this is typical of character displacement. All right, so let's take first the simplest case. Species are identical in their growth strategies. So they have the same theta V here, and that's given by 1 dashed line. Now, if there were no competitors here, each species, if a species were growing by itself, it would evolve to match this particular value. But in this particular example that I've given right here, I've got 2 species, a blue and a green. And if we track their germination strategy, you can see that they diverge over evolutionary time. And this is an example of character displacement. In the model, what's required here is that the costs of germinating at the wrong time are less than the fitness reduction due to interspecific competitive interactions. And that makes perfect sense. If we flip it around the other way and we make those costs large, or if we increase the costs, or we reduce the strength of competition between species, we can get collapse of that divergence, and the 2 species essentially do the same thing, and one of them will go extinct in this particular model. We can theoretically uncover what's going on here by deriving a selection gradient. And I'm not going to go through all this. I just want to note that we've got 2 terms. This is the density independent selection term, which reflects within species trait fit. So whether these 2 traits fit together or whether they don't, whether the germination and growth strategy are matched. And then there's also the association between the germination response and competition, and that's the action of character displacement. All right, so to summarize what I've shown you before, the two species had the same growth strategy, so their difference was 0, and I showed you two outcomes. One where there was strong competition, we got lots of divergence, lots of differences that evolved between species in their germination strategies, and one where the costs were quite large, there was weak competition, and so we got no divergence at all. So what happens when we allow the growth strategies to differ between the species? Well, this is what it looks like. We get lots of divergence, and the most important thing about this figure is that very small differences between species and their growth strategies jumpstart adaptive divergence between them in some other character, in this case, germination. So we've got some niche differences between species initiating character displacement in other traits. All right, so to summarize what all this means, what I just said here, differences between species in one trait can influence divergent selection on another trait, and that shouldn't be too surprising. What's important here is that there's potential for within organism fit to be important. Some trait combinations reduce fitness independent of the action of competition. So I showed you that. But also, and so that can inhibit character displacement when species are very similar in this trait, but it can also jumpstart and facilitate character displacement when species differ in this other strategy. So instead of looking at selection on beak size by itself, maybe we should look at selection on beak size and shape, conditional on maybe leg length or leg shape or something like that. So I'm not an ornithologist. That probably doesn't make any sense, but just as an example. All right, and the implications for this is that some seemingly similar species pairs may show very disparate divergence responses in similar environments. And I think what we can take away from this is that some niche differentiation can act as fuel for further niche differentiation in other traits. And that these kinds of trait interactions in competitive systems are probably important in the field of ecological syndromes. So in annual plants, we do think that germination and plant growth strategies are somewhat tied together for this reason. And finally, I'd like to say that the storage effect appears evolutionarily robust given strong competition or when competition is determined by more than one trait. So that's nice. That's a nice result. In future studies, we need to consider other evolutionary models, perhaps more than 2 species, since I told you there are 50 that are out here, and the action of other important ecological players like some seed predators. But we'll save that for later. So I'll take any questions if you have them, but first I want to thank my collaborator, Peter Chesson.
Sorry, thank you very much for that nice talk.
So this is a question from Mark. Great talk. In the model formulation, are the germination and growth strategies tied to any particular environmental features, or is the formulation of the model simply based on the covariation among species, regardless of the environmental drivers?
Right. So there's no specification of any particular environmental variable in the model that links germination and growth, but we do have kind of a phenomenological term that represents the amount of information at germination about future growing conditions. So this can be a lot of things in the field, like the time of the year when species germinate. So if you germinate early, there's probably a good chance you're going to experience a drought because rain comes sporadically in the desert. If you germinate late, then your seasonal length is pretty short. So there's some information in there. We don't know exactly what those are, but we modeled it phenomenologically.
We have a question from Carol. It says, cool link of plants and storage effects to character displacement theory. Perhaps this is just a language issue. I understood that the correlation of -1 is not uncorrelated. It is strongly negatively correlated. What one species is doing is highly predictive of what the other species is doing.
Yes, that's right. And that's when you get the strongest storage effects in that case. And interestingly, I didn't show this, but when the species are doing that, when they're completely negatively correlated in their germination fractions, they're often they appear very poorly fit to the environment. One or the other does.
Great. Yeah. I think we need to move on to the next one. Yeah, I have 239 on mine. Yeah, 240. There we go. Our next speaker is Kelsey Lyberger, Clonal Coexistence and Turnover in Daphnia, the Role of Resources and Random Chance.
Unmute myself, get this going. All right. My name is Kelsey Lyberger. I'm a 6th year PhD student at UC Davis. I'm going to talk to you. Oh, everyone's face is in the way. Let's stop that. About clonal coexistence and turnover in Daphnia, the role of resources and random chance. So Nick set me up really well by explaining that one of the major goals in our field is to understand what explains species diversity. So part of this has been a long history of debate between neutral models on one side and niche and resource-based ones on the other. And so I'd like to apply these same two ideas, but to the question of what maintains diversity within a species. So this is especially important given that we now know that genetic identity and diversity at that lower level can have important effects on other ecological processes. And I argue that one of the best ways to answer this is using clonal organisms. Like shown here, we have our Zostera eelgrass, solidago plants, and my favorite, Daphnia. And a follow-up question is what drives turnover in clonal composition? Again, having these two ideas of could it be all random or are biotic and abiotic variables driving turnover in a predictable way? So sort of an overarching goal of this project is to assess the roles of random chance and seasonal selection in explaining coexistence and turnover among genotypes of Daphnia. And to do this, I've used three approaches. First, a set of field mesocosms, also some lab life history assays, and third, some long-term monitoring of a natural population. So the first, using mesocosms, I aim to test what extent clonal dynamics of Daphnia in a pond are deterministic. And I came across this very convenient system in a pond at UC McLaughlin Natural Reserve. And when I first sampled the Daphnia population in spring of 2018, it really only consisted of two dominant clones in this pond, the blue clone and the red clone, and I'll use those colors for the rest of this talk. So here you can see them battling it out in the pond. And so I set up a set of 50-liter mesocosms that are floating in the pond. I filtered out all the other zooplankton from the water and then added in either a mix, a 50-50 mix of the red and blue clone, or either just red alone or just blue alone. I then monitored clonal frequency changes over the next 6 weeks, looking for any parallel changes across these replicate populations. So to start, we'll look at the more interesting treatment, our 50-50 mix. And what's really exciting is that we saw in many of these populations that the red clone took over. And in almost all, the red clone is increasing in frequency. Then for our blue clone, we had what I call a happy accident in a few of them, which is where one or two of the red clone got accidentally introduced during sampling. And so you can see in these pure blue, the red clone increased in frequency in these bottom two. Then in the pure red enclosures, again, we had some happy accidents down here, but when this blue clone entered, it was never really able to invade. And overall, I see this as a relatively predictable outcome, that something about this environment is favoring the red clone. So the next step was trying and understand why red may have been favored. And so I tested whether two clones exhibited differences in their ability to utilize resources and withstand starvation. And this is not all my idea. In fact, this trade-off in resource exploitation exists among species of Daphnia. So here we see in this graph from Tessier et al. 2000 that a graph of growth rate over mean resource value with a line for each species of Daphnia. The species represented by stars here has the highest growth rate at high resource values, but is expected to have the lowest growth rate at low resource values. Whereas this unfilled circle species is relatively insensitive to changes in resource values. So if we apply these ideas about differences among species to within Daphnia, we might expect this from my two clones, where the red and blue differ in their sensitivity to changing resources. So I ran a small lab experiment where I exposed the red and blue clones to either high or low resource values and measured a set of their life history traits. So what I found is that the red clone has the higher growth rate at high resources compared to blue, and they both have about the same growth rate in low resources. Then our red clone has an earlier age at maturity at high resources than the blue clone, but a later age at maturity at low resources compared to blue. So we do see these crossing reaction norms where it seems like the red clone is superior at high resources and the blue clone is superior at low resources. So from what we know now, we can start to make predictions about what might be happening in terms of turnover in this pond across seasons. And because my mesocosm experiment happened during the summer and maybe resources are higher during summer, we might expect to see the red clone dominating during summer and the blue clone dominating during winter. So the third part of this project was to test whether seasonal changes create consistent patterns in turnover. And I did this by documenting clonal composition monthly for over 2 years. And so here's what I'm going to show you is the composition over these 2¼ years, where we might expect during these yellow spots of summer to see more red clone and blue sections of winter to see more blue clones. So it's not exactly what we predicted, but I guess I want to show you a few things that happened over time. The first is that during the summer when I set out those mesocosms, the red clone did increase in frequency more than the blue clone. But clearly this appearance of this green clone overshadowed that. Then in winter, we did see that the blue clone dominated, and the next summer, the red clone took over. But we never again saw the blue clone. And of course, there are many other clones hanging out in the population. So there's some evidence for seasonal selection here, but it's much more complicated as new clones are appearing and others, like the blue clone, disappeared. And here's where my talk becomes especially speculative. So just as we've seen that the red and blue clones aren't existing in a vacuum, they're not interacting in a bubble by themselves. There's many other clones. Neither is Daphnia pulex as a species. There are many other things happening in the zooplankton community. So here's a plot down below of the composition of the rest of the zooplankton community with our focal species, Daphnia pulex, in purple. And I want to point out a couple of things. The first is that right around the time that our blue clone disappeared, we have the appearance of a new Daphnia species, Daphnia dentifera, which perhaps outcompeted our blue clone. But also at the end of every summer, Daphnia pulex goes through a bottleneck. And so it could have been during this 2019 bottleneck that led to the extinction of our blue clone. So what did we learn? That from our mesocosms, some of these changes in clonal frequency are predictable, and that perhaps it has something to do with the environment and resources. In second, we learned from our lab assays that trade-offs in resource exploitation can explain coexistence between these two clones, but that in a natural population, it's going to be the combination of both chance and seasonal drivers that are needed to explain clonal coexistence and turnover. So I'd like to thank my funding sources, my advisors, Sebastian Schreiber and Tom Shaner, committee members, the several undergraduates who helped with the molecular and field work, as well as those who taught me to work with Daphnia and microsatellites. So I'll leave you with this nice video and ask for questions.
We have time for questions.
We have a question from Becky. Is there any role for parasites here? I know that some of the Daphnia parasites also have a seasonal cycle.
Yeah, I have not seen parasites in these Daphnia populations, at least ones that I have noticed under the scope. Okay.
Annika asks, are certain clones maybe just more susceptible to certain parasites? Another parasite question.
Yeah, I think I need to talk to Megan Duffy more about when parasites show up.
Casey has a question. It's, hey, Kelsey, great talk. I was wondering if there's a relationship between clonal composition and total Daphnia pulex abundance.
Yeah, so I think that, that here it could be that these bottlenecks in Daphnia abundance are explaining some things about clonal composition. But I think I would need years more data to know what those relationships are.
So I have a question. What are the predators in this system? What's out there preying on in these ponds? Are there fish? Are there amphibians?
Yeah, so the big one is probably fish. There are bluegill in this pond that have been introduced, and so they obviously have a much higher activity at certain times of year. But I haven't noticed differences in, for example, size or coloration of these two big main clones. Oh, and the other predator is Chaobarus. So yeah, they're sort of being beaten down both at the top of the water and bottom.
Okay. Becky, do these clones differ in their propensity to undergo sexual reproduction?
That is something I don't know, but what I have noticed in this population of Daphnia is that maybe it's because it's California and it never really becomes unfavorable, but they don't produce many resting eggs really ever. And so even at high densities and even in the dead of winter, I rarely see any resting eggs, but it does seem like in, what is this, February or March of last year, there are like a handful of new clones, and that suggests that perhaps there was emergence from the egg bank.
If there are no other questions, we have sorry, I'll shut up.
Are these clones their clones, the new clones, are they generated from the existing clones, or are they possibly coming in from the outside, being introduced?
Yeah, so I don't know that for sure, but I do have evidence that they're very similar to the other clones in the population. So it's possible they're coming in from outside, but if it were, that source population would have to be pretty similar.
Well, I'll ask one more myself. So I think of that part of the world as being pretty dry a lot of the time. But there's bluegill in there, so clearly this is a permanent pond. What's the water source, or is it fairly deep so that it's fairly resistant to drying out?
Yeah, this is a dammed pond at the reserve. So the water level does fluctuate throughout the year, but it's at least 4 meters year round.
That would do it.
Well, thank you for listening.
Yeah, we have about a minute before the next talk starts here for folks. When I teach class, I wait for the time to actually click over. I'm like, oh, it's time to go. Sorry. Okay, our next speaker is Kristen Nolting. The title is Coexistence and the Evolution of Interspecific Variance under Interspecific Competition.
Sorry, just getting this set. All right. Can I get someone give me a thumbs up that you see my slides and not my presenter view? Okay, cool. All right, well, and get this up here. I'm a new postdoc at the University of Georgia, but today I'm presenting some of the results from my dissertation research, which I recently completed at the University of Connecticut, investigating the drivers of plant diversity in the Cape Floristic region in South Africa. And before diving in, I want to amend my title to reflect that I'll be sharing some of the empirical work that motivated my interest in developing a model to explore the evolutionary dynamics of trait-mediated competition. And then I'll finish up with some of the preliminary work I've been doing to investigate these questions using an individual-based simulation model. So it's been fun hearing the talks so far, I mean, throughout the conference, but especially in this session today, because I think most of us would agree that one of the most fun things about being an ecologist is getting the opportunity to explore species diversity in natural systems, and not just species diversity, but diversity of form and function. And so the stunning diversity in traits that we see is not only beautiful, the traits of an organism help us understand how it is interacting with its abiotic and biotic environment. So much of my research focuses on understanding how organismal trait diversity can inform species diversity at different ecological and evolutionary scales. And so for my dissertation, I was really lucky to be able to investigate these questions in plant communities in the Cape Floristic region in southwestern South Africa. The CFR, as it's called, has over 9,000 species in approximately 90,000 square kilometers. 60% of these species are endemic, and more than half of the species are representative of 33 species-rich lineages. So given its extraordinary species diversity and high degree of endemism, the CFR is recognized as a biodiversity hotspot, which is kind of cool given that it's not located in kind of the equatorial tropical belt. So the diversity in this region has resulted from really dynamic ecological and evolutionary processes that I can't get into today. But for today, I want to highlight that the plant communities I focused on for my dissertation are what we see or saw in this picture here, feignboas habitat. And this habitat is defined by relatively low rainfall, nutrient-poor soils, and importantly, naturally recurring fires that come through every 12 to 15 years. And specifically, I studied feignboas communities dominated by members of the Proteaceae family. So here's just a small subset of the species in the genus Protea that are endemic to the region. And I mean, clearly they're beautiful, right? But as an ecologist, something that I found especially striking about feignboas plant communities, and especially those dominated by Proteaceae, is that within a fairly restricted local community, you can find 4, 5, even 6 or more different species within the same lineage co-occurring. And so wouldn't we expect species from the same genus to be relatively ecologically similar to each other, given their shared evolutionary history? And then might this ecological similarity then make them more likely to compete for shared niches? If so, how is local diversity maintained? Well, species in the Proteaceae family show a high degree of trait variation, as we saw with their floral diversity, and also with respect to their vegetative diversity, which we see here. So this is just a small subset of the species that I worked with, but there's tremendous diversity in leaf shape, size, overall growth form, and even fire adaptation strategies. But these trait differences, we don't necessarily know if these trait differences help mediate the negative effects of competition when species co-occur. Alternatively, we might expect that co-occurring species have kind of a shared regional or kind of local adaptation to the environment, and so might actually have relatively similar trait values that allow them to persist in their shared environment. And so For the last chapter of my dissertation, I kind of focused on this dichotomy of whether or not we'd expect co-occurring species to be similar or dissimilar in their traits, and if trait dissimilarity itself could help explain kind of interactions among individuals in local neighborhoods. So the first question that I'll talk about today was looking at whether or not variation of traits of a focal individual have fitness consequences for that individual. So I'm calling this a trait optima effect. And then kind of the alternative side of that is whether or not trait dissimilarity among neighbors have fitness consequences for the focal individuals, the trait dissimilarity effect. So a quick note before we get to the study design, I mentioned this is a fire adapted ecosystem, and that's important. And so what this means is that fires come through every 12 to 15 years. And when it does, the adults release their seeds from a persistent seed canopy and then die. Note that this also allows me to get lifetime reproductive effort on a long-lived species, which is pretty cool. And then when the rains come, seedlings come up at about the same time in mass, and this results in dense stands of even-aged individuals as adults. And so many of these stands are monospecific, but often there will be a second Proteus species hanging out in a neighborhood of the first species. In sum, this allows me to compare trait and trait dissimilarity effects in neighborhoods that are largely conspecific versus heterospecific. Okay, so with that background, I'm just going to show you a general sampling design and a general model framework without getting into the gory details. But I first in the field identified local neighborhoods with either within either conspecific or heterospecific stands. And then within these neighborhoods, I measured the size and height of all individuals in that neighborhood, and I collected leaves from each individual for trait sampling. I also counted the total number of seed heads and inflorescences on the focal individual in each neighborhood as a proxy for fecundity of that focal plant. And with this data, we were able to evaluate the effects of trait value and trait dissimilarity on the fecundity of the focal plant, taking into account variation in neighbor density. So I'm just going to dive straight into the results. First, I want to describe the predictions we had with respect to my first question, which asks whether or not variation in traits of the focal individuals themselves is associated with that individual's fecundity. So from previous work, I know that many of the leaf traits that I sampled for the purpose of this study are related to fitness in many proteas. So I did expect to be able to detect these fitness associations once again in this study. So for example, in my cartoon figure, I see a negative association between trait 1 and fecundity, this negative effect, but a positive association with the second trait measured, this positive effect here. And I'll note that the effect sizes here, the mean effect sizes, are represented by the black dots, and then the bars represent the uncertainty in those estimates. With respect to comparing conspecific versus heterospecific neighborhoods, I didn't really have any a priori expectation that the association between a trait and fitness would be dependent on whether an individual occurs in a conspecific or heterospecific neighborhood. So I thought these effects would be more or less the same. So looking at the trait optima effects, this is first looking at neighborhoods that were largely conspecific. The results here are presented just as we saw, but now I have the 5 traits here on the vertical axis. And we can see that of these 5 traits sampled, 2 have effects that seem to be important. Specifically, we have leaf dry matter content has a negative association with fecundity, but lamina density, that trait on the bottom, is positively associated. And these other traits have effects that are indistinguishable from 0. When we look at heterospecific neighborhoods, however, we see that those top 3 leaf traits are negatively associated with fitness, and now leaf dry matter content and lamina density show opposite relationships with fitness. I admit that this was a little surprising, but intriguing, and I am currently exploring this a bit more. But I just want to say, in sum, we do have evidence that kind of the absolute value of the traits themselves matter for the focal individual. So evidence of this trait optima effect. So second, let's look at the effect of trait dissimilarity between our focal plant and its neighbors. If trait dissimilarity mediates competition, we'd expect to see a positive association between trait dissimilarity and fecundity of the focal individual. So in other words, the more different you are from your neighbors, the better you do this positive effect. If, however, competition is greater among conspecifics, as might be predicted for species coexistence, I would expect that the magnitude of this trait dissimilarity effect to be greater in conspecific neighborhoods than in heterospecific neighborhoods, the magnitude being greater. So in conspecific neighborhoods, I just want to orient you again. The vertical axis here represents the traits, but in this instance, this is the average dissimilarity of the focal individual with respect to all of its neighbors. So it's the effect of that trait dissimilarity. And we see that in several traits, being different from your neighbors is good in conspecific stands, but there's also this evidence that being dissimilar from your neighbors in other traits is bad. So these negative associations could indicate that there's just some optimal trait value for that local neighborhood, which is consistent with the trait optima effects we just saw. But we do see that some trait dissimilarity seems to be important. In heterospecific neighborhoods, we also see this mixture of positive and negative dissimilarity effects, but in general, the uncertainties in these estimates are quite large. So this makes it difficult to make inferences from these results with confidence. All right, so conclusions from the field. There are fitness consequences associated with trait variation of focal plants, and this depends on neighborhood type. Second, being dissimilar in some traits is good, and in other traits being too dissimilar is bad, and this might be due to this local trait optima. But kind of the big take home for me from this massive study and a lot of trait collection and quantification is that the trait variation within neighborhoods could be important for ecological interactions. And so specifically, this is trait variation measured at the level of individual plants, which is kind of cool. So even though this is a cool result, and I'm working to finalize this manuscript for submission, I am still left feeling a bit frustrated as this study really captures the outcome of ecological interactions in one moment in time in the long history of ecological and evolutionary dynamics in this community. I really want to be able to explore the role of ecological similarity and dissimilarity in driving evolutionary change, and to investigate if the evolution of trait variance itself is important for ecological dynamics and outcomes. But to do this, I can't rely on field studies with plants that live 10+ years before reproducing. So at the time I was wrapping up my dissertation last spring and considering these unanswered questions, I was participating in a seminar course at UConn led by familiar faces, Mark Urban and Dan Bolnick. And the format of the course was actually structured similar to AMNAT-style presidential debates, in which we would read papers and take sides on key questions with respect to ecological and evolutionary perspectives. And while it was fun for me to pretend I was in sunny Asilomar while in cold Connecticut, I'm highlighting this as this course really pushed me to think more critically and read more broadly about these questions. It also gave me a bit of boost of the confidence I needed to dip my toes into the waters of theoretical eco-evo and simulation models. So the model that I started to develop was the outcome of a paper I wrote for this course. So big thanks to Mark and Dan. To start, I'll describe a verbal model that I'm using as a framework. So we can imagine a hypothetical community with two species, perhaps similar to fambose communities I studied for my dissertation. Each species has a starting abundance, and together these abundances equal the carrying capacity for the environment. Individuals within each species also show variation with respect to some continuous trait, and every individual in the community interacts with every other individual, both conspecifics and heterospecifics, with respect to this trait dissimilarity. So there are trait-mediated competitive effects such that individuals that interact with other individuals that are kind of distant from them with respect to their trait interact less intensely versus individuals that might be more similar in traits are going to interact more intensely. There are also interspecific competitive effects that are trait independent. So essentially, I think of these as the effects that are not explained by the trait of interest. And also that there is some phenotypic optimum for each species, represented by these thetas here. And individuals that have phenotypes that are distant from their respective optima incur greater fitness costs. Second, we want to be able to quantify the phenotype of each individual based on a genotype. So for my model, I'm keeping it really simple and assuming each genotype has 40 loci, it's biallelic and completely additive. So with this phenotype and understanding of ecological interactions, we can quantify the fitness of each individual in the community with a fitness function. All right, so this is the point where I kind of finished up the course, had the verbal model set in my mind, felt pretty good about it, but needed to be able to define a reasonable fitness function that could be incorporated into kind of the eco-evo simulator that I was developing. So in October, I was skimming the table of contents of the just accepted articles in AmNat and saw this really cool and really timely paper in which the two authors here investigated the ecological consequences of interspecific variation in coevolutionary systems. So the authors explored multiple types of coevolutionary systems here, but for my interest, I was especially excited to see a clear description and presentation of a fitness function that I could use for my simulator. Highly recommend checking out the paper. It's a really, really cool resource. So for those of you who are familiar with these types of models, you'll notice that this is essentially a two-species Locke-Volterra model of competition with respect to a continuous trait X. With the negative effects of trait-dependent competition and dissimilarity from a trait optima are both described using Gaussian functions, and the trait-independent effects of heterospecifics are defined by competition coefficients.
Kristen, there are 4 minutes.
Great, thank you. The fitness of focal individuals is kind of the summation of these negative effects balanced by some intrinsic rate of increase that's specific to each species. So with my fitness function, I can now quantify the fitness of all individuals in my community and then determine who survives based on its species-specific relative fitness. And then after this bout of viability selection, we have survivors of each species that are then allowed to reproduce. There's random mating within species, offspring inherit intermediate parental genotypes, and offspring are added to the community until the number of individuals in the community reach the carrying capacity. But note the abundances can change. And then with the offspring, we can translate individual genotypes to phenotypes to get our new community at generation T+1 with the new phenotypic distributions for each species. We can repeat this process for a set number of generations and observe evolutionary dynamics. So for example, this is just one kind of output from one run with one set of parameter values. And we can see how the phenotypic distributions of the two species change from generation 1 to generation 300, and then also how the relative fitnesses of each species based on their phenotypes shifts through time as well. We can also look at how the demographics of each species change through time. So as the traits are changing through time, we also see shifts in the relative abundances of species 1 plotted here. So as you might expect, the way in which these communities change is very different based on the way we parameterize the model, and I'm currently working to summarize and generalize this output with respect to several ecological scenarios. And I'll add here, this is work that I'm doing with my friend and collaborator, Stephen Delisle, who's been just awesome to work with on this. Ooh, so that was a bit of whirlwind. I'll stop here and will thank the folks on this slide, especially huge thanks to my PhD advisor, Kent Holsinger, and I'll take questions if there is time. Thank you.
Nice job. I think there's time for a question.
Okay. Yes. This is about the trait dissimilarity idea is pretty cool, but does it rely on the other species really being a competitor? Thinking about my own systems, there might be other species nearby that are genuine competitors where dissimilarity might be important, but there might be others close by that are irrelevant. Might this account for some of the variability in the heterospecific dissimilarity results?
So with respect to like the empirical data, I would say yes, there's probably differences in the degree of interspecific competitive effects. It sounds like I cherry picked this system, and I kind of did. I cherry picked this system to ask these questions because the communities where they co-occur, it's largely the Protea species that have similar growth forms and seem to be interacting most intensely. There's other grasses, there's other annuals and geophytes. Temporal variation is very important in these systems, but at least kind of at the time scale at which these populations are interacting, I would guess that these would be the species that would be most intensely interacting with each other, whether or not they're competitive. I think I have evidence of it, but I'm still working on, I will say I have density data, so I'm looking at the density effects in the neighborhoods as well. But those results are still not conclusive.
Thank you very much. I think we need to move on to the next speaker. Our next speaker is Linyi Zhang. The title is Context-Dependent Reproductive Isolation. Host plant variability drives fitness of hybrid herbivores. Linyi, I think that you're muted.
Oh, whoops.
Yeah, do you want to start over? Sorry.
Oh, very sorry. Okay. Yeah, okay, I didn't see that. Okay. So I'm going to introduce myself. My name is Linyi Zhang, and right now I started a postdoc with Zach Gombert at Utah State University, and today I'm going to share my findings during my PhD work at Rice. So there's an increasing awareness that rather than a static parameter, reproductive isolation between two species varied across time and space. Most of these studies have focused on the polymorphic genetic incompatibility that leads to the variation in postzygotic reproductive isolation. However, there's only a few examples showing how changes in the environment could also alter the strengths of reproductive isolation. One classic example is in cichlid fish. When water clarity decreases due to eutrophication, the sexual isolation between the fish species collapse, leading to the collapse of species boundaries. The similar pattern is also observed in Darwin's finches. When the El Nino event occurs that changed the seed size distribution on the island, this improves the hybrid fitness, therefore facilitating the hybridization going on for over a decade, again blurring the species boundaries. All of these examples have stressed the importance for us to understand both the pattern and the cause of variation in reproductive isolation. Given the potential importance of the environment in driving variation in reproductive isolation, it is surprising that in the field of ecological speciation, which mainly focus on the role of divergent roles in generating reproductive isolation, researchers have rarely considered the effect of heterogeneity within each divergent environment and how that could affect the magnitude of reproductive isolation. We argue that herbivorous insects would be the ideal study systems to address such knowledge gap. This is because many of these insects are highly specialized on a few host plant species, making the source of divergent selection easy to identify. And what's more, there's lots of literatures showing the heterogeneity within each host plant species, which means that individual host plant varied a lot in terms of the quality. And for our study, we specifically focus on a gall-forming specialist, bee treaty, that specialized on two sister live oak species, Quercus virginiana and Quercus geminata, which referred in QV and QG for the rest of my talk. And these two species have geographically broadly overlapped in southeastern United States, where molecular work have showed that these sympatric lineages have formed distinctive genomic structure. As you can see by this bar plot, where each colored bar is representing one individual. And you can see some orange individuals mixed in the QG host associated population, suggesting there's some migration going on between the lineages. However, very few individuals with a mix of two different colors, suggesting there's very little genomic admixture. And corresponding with a distinctive genomic structure we see, our collaborative work has found that these sympatric lineages have formed near complete prezygotic reproductive isolation, accumulated by multiple reproductive barriers, including temporal isolation, habitat sexual isolation, and a more rarely described barrier, which is reduction in immigrant viability and fecundity. Furthermore, previous studies have also found there's a great variation in individual host quality, leading to the patchy distribution of the galls. All of these previous knowledges have set up the perfect stage for us to test the following two questions. First, is there any form of post-zygotic reproductive isolation, particularly hybrid inviability, exist? And second, does individual trees contribute to the variation in the magnitude or direction of hybrid fitness? To test that, we went to collect root galls that contains both males and females across this distribution range, where we also prepares individual potted plants that grows from seeds collected from multiple sites across Florida. We then bagged multiple individuals of different cross types on individual potted plants. This including pure crosses where males and females come from the same host plant and also feeding on the native host plant, and hybrid crosses where males and females come from different host plant. We then quantified the offspring fitness by measuring the proportion of host plant hypersensitive response. So when females oviposited the eggs, they created a permanent brown scar on the side vein of the leaves. This enabled us to quantify the amount of oviposition events. And the later development of the larvae could induce the plant immune response that kills the cells around it, therefore reduce the chance of gall formation. In total, we have conducted over 300 crosses on 66 individual trees and counted over 50,000 oviposition scars. So for question number 1, is there any form of hybrid inviability? Here on the y-axis is the offspring fitness, where the x-axis is the different cross types reared through different host environment. What we found is that only one hybrid cross direction, when female QV mated with male QG, have significant lower fitness than pure cross, but only on QG host environment, not on QV. Because this hybrid, the genomic composition of this hybrid crosses should be the same, this environmental dependent hybrid inviability also suggests that ecology rather than genetic incompatibility is the underlying mechanism. We then to test whether it is the individual trees that drive the variation in hybrid fitness. And to test this, we conducted a generalized linear mixed model where we with three different random structures. Our null hypothesis is not including individual tree as random effect, where our model 2 included individual tree as a random intercept. A significance of this random structure would suggest that the offspring fitness varied across individual trees, regardless of cross types. Where the third model, we included individual tree as a random slope over cross types. A significant effect of this random structure suggesting that differences differences in offspring fitness between resident and hybrid varied across in vitro trees, suggesting in vitro tree variation drives the direction and relative strength variation in hybrids. And what we found is that indeed the model 3 is the best fitted model. And we further calculated the total amount of variance explained only by the fixed effects, which is a marginal R square showed here, compared with the variance explained by the whole model, which is a conditional R square showed here. You can see that by including in vitro trees a random slope, the amount of variance explained increased by a magnitude of almost 5-fold. Why the model comparison result suggests in vitro tree effects on hybrid fitness? It's acting like a black box. So to further validate this point and visualize the variation across in vitro trees, we compared offspring fitness of resident and hybrid on the same tree. Here, the negative slope would suggest hybrid viability, while the positive slope would suggest hybrid vigor. And no significance in the slope would suggest no differences between hybrid and resident fitness. And here's a visualization. You can see there's a lot of variation in the direction as well as the steepness of the slope. And to further quantify this variation in the slope, we conducted a generalized linear model. We included individual trees with multiple replicates of resident and hybrids. And here in this figure, the y-axis is a list of 25 individual trees, and the x-axis is a regression coefficient within each individual tree. The negative value indicate hybrid inviability, while the positive value indicate hybrid vigor. And here's what we found, and I'll walk you through what is going on in this figure. So the black diamonds represent the mean regression coefficient, with black line indicate the 95% confidence interval. Any overlap of the black line with the value 0 indicate no significant differences between hybrid and resident fitness. And the colored dot is the pairwise differences between each replicate of hybrid and resident. And here the histogram shows the distribution of the value of regression coefficient, suggesting there's a lot of variation across in vitro trees. So 7 trees exhibit hybrid inviability, while 9 trees exhibit hybrid vigor, and 9 trees exhibit no significant differences. The latter 2 will all potentially facilitate the gene flow between the lineages, constrain the speciation process. So to summarize, what we found is that first, there's a lack of intrinsic hybrid inviability between these host-associating lineages despite a pretty high degree of genetic distance. This could be because we're only measuring the hybrid fitness at the early life stage. But it could also be that the relationship between the hybrid inviability and the degree of genetic distance is unique in the haploid-diploid system. We're happy to find that the research led by the Linyi group also find a similar pattern in their haploid-diploid softfly system. And you're welcome to go to see their read their paper to see the potential mechanisms. And one possible explanation is that the deleterious alleles are purged much faster in the haploid males. And second, we find that their individual tree variability drives the variation in hybrid fitness, causing this context-dependent hybrid isolation. We argue that this context-dependent reproductive isolation is critical for us to predict the speciation dynamic. And you can imagine individuals using intermediate environments as a stepping stone to migrate from one population to the other, potentially constrain the speciation process. And lastly, even though our study presents one of the few examples of context-dependent reproductive isolation, we argue this should be a prevalent scenario, especially in the context of ecological speciation. This is because it is really hard to imagine any environment are entirely homogenized entity. And with that, I'd like to thank everyone in Scott Egan's lab that helped with my project, as well as all of the societies that partially funded my dissertation research. And with that, I'd like to take any questions, and you can also contact me via my email or Twitter handle. Thanks.
Nice job. We have time for questions.
Nothing's come through yet.
Well, I just want to say that we're also interested in developing some model to test the effect of how heterogeneity within the environment and the spatial configuration of it could promote or constrain the speciation process. So if any people with modeling expertise, especially in the individual-based simulation model, you're welcome to just contact me. Thank you.
Excellent. So were your hybrids all were they? I might have missed this, and you might have said it. Were your hybrids in the F1 stage?
Yes, it's F1.
Yep. Yes. So what do you think would happen if if you did a back cross, or the F2s where you're more likely to get, I know, the various genetic incompatibilities?
It could be, but I just want to note that this system is a non-model organism, and also it has very complicated life cycle. It has both sexual and asexual generation. So it's just like experimentally, it's almost impossible to produce the F2 hybrids in the lab. Yeah, but that'd be very interesting because it's totally possible that would happen.
Did you? I think you did say this at the end. So it was also the case that you were looking at the survival in the early life history stages, like you weren't able to. Yeah. So like look at the fecundity of the females, or the fertility of the males, or any of that.
No, so here the other thing. So our hybrids are actually asexual generation, so we only have females. But there's our experiment goes, and unfortunately we just don't get enough gall formation that produce enough hybrid individuals that even be able to quantify the female fecundity or even the gall formation success, because they're just it's just the imperfect of field experiment, I guess.
Cool.
If you think of anything, you can send it to Linyi on the chat or for email, and she'll answer your questions.
Sent my email to the chat? Is that what he said?
I think that would be good. I think they can also get it.
Yeah, sure, that'd be great. Yeah, because I really want to develop some modeling stuff, and it'd be nice to have people collaboration.
Okay, I think we can go ahead and get started on this next one. I think we're close enough to 3:40 here. or 440. Our next speaker is Benjamin Freeman, and the title is Faster Evolution of Song Discrimination Leads to Faster Diversification in New World Passerine Birds.
Thanks. That was the title before I ran it through the clickbait translate filter, which is a good one. So the problem I want to talk about today is the tremendous variation that we observe in how fast different lineages on the tree of life produce species. And as an example, and it's a problem because we observe huge variation and we're often unable to explain almost any of it. As an example, consider New World birds, a group dear to my heart that I'm going to tell you about today. Well, they have a 30-fold difference in the rates at which they are producing species at present. Some lineages producing new species 30 times faster than others. We really don't know why. One hypothesis is that we can explain this variation if we knew how fast populations evolve reproductive isolation. I'm going to term this the RI hypothesis for reproductive isolation, that some lineages have populations that evolve reproductive isolation faster, and those are the lineages that have faster, longer-term speciation rates. And this is an intuitive idea because speciation, after all, is often defined as the evolution of reproductive isolation. But the one study that tried to look at whether these rates of reproductive isolation over relatively shorter timescales and rates of speciation measured over longer timescales found no such association. And this was looking at both a group of birds, of warblers, and also Drosophila. And so an alternative hypothesis is that reproductive isolation, these rates of evolving reproductive barriers, just essentially don't matter for speciation rates. And a way to phrase this is that they're not a rate-limiting step in speciation. And this could arise because, at least in birds and probably more broadly in vertebrates and other texts as well, speciation can be usefully conceived as a three-part process. First is range fragmentation, that a species with a large range the range becomes fragmented for one reason or the other. The point is that differentiated populations are formed. This is a demographic factor. The second is reproductive isolation evolves between at least some of these isolated populations. This could be ecologically driven, this could be mutation order processes. And the third is that these species, once formed, expand their ranges. Again, this is necessary to restart the cycle. If species once formed only lived in smaller and smaller ranges, future bouts of speciation would be curtailed. And so that puts an emphasis on ecological factors. And so in this kind of way of thinking about speciation, reproductive isolation is only one of three steps. There's no does not have to be the rate limiting step. And we have some evidence that the demographic factors, the rates at which differentiated populations are formed, and ecological factors like rates of range expansion seem to be important for these longer term speciation rates with the citations I've highlighted here. And so with that buildup, that introduces the study I'm going to tell you about today, where we set out to test the association between the evolution of a reproductive barrier, so one component of reproductive isolation, and we looked at song, and then speciation rates in New World passerine birds. And we studied song because birds sing to attract mates, and they use song to choose mates, and it's traditionally been considered to be a trait that's important for reproductive isolation. As Darwin expressed this viewpoint by saying, everyone knows how much birds use their vocal organs as a means of courtship, which I like this quote partially because of the rhetorical device that I think from now on when I make statements, I'm just going to preface it by saying everyone knows. It's hard to argue with that. But so then for this study, looking at song evolution, the predictions of the hypotheses I just laid out is that for the reproductive isolation hypothesis, we predict a positive association between trait evolution, that's how fast the songs evolve, and speciation rates. And then the alternative hypothesis predicts no such association. And the way that we studied song evolution was in the context of this, of how speciation is initiated in birds, where these reproductive barriers evolve between geographically isolated populations. And so we studied 175 taxon pairs, where each taxon pair is a pair of related populations, as illustrated by this, this mini phylogeny of a blue species and an orange species. And these two populations live in different geographic regions. And I should clarify that they're two populations. Some of them are classified as distinct species. Sometimes they're classified as different subspecies. And if we were at Asilomar as we woke up this morning and strolled along the Pacific admiring the waves and the birds and whatnot, we would have clearly enjoyed seeing this blue species. And the question then is how to measure the degree to which these two populations within each taxon pair, how to measure their song divergence. And this is tricky because song is a super multi-dimensional trait. And in the context of the study, what we wanted to study song as a reproductive barrier. And so what we ultimately decided was to ask the birds themselves whether the song kind of was likely to constitute a reproductive barrier or not. What does this mean? It means that we did a hellacious number of playback experiments, 2,339 in the Pacific Northwest on lots of taxon pairs there, where I'm based now, in the Southwestern US, in Central America, and in the tropical Andes. And this is a hugely royal we. Graham Montgomery in particular was crucial in doing this field work, which was fun, but extensive. And each playback experiment, what it did was it simulated secondary contact between the populations within the taxon pair. So we'd go out and we'd find a territory of, say, an individual from the blue population. And we'd do a playback experiment with two treatments. One of the treatments was local song. We'd broadcast song from its own population. And a territorial bird confronted with song from its own population, it gets pissed off. It gets angry. And we can measure that anger by its behavioral response. Basically, they'll approach the speaker, they'll look for this intruder and try to kick it out of their area. And the other treatment, and we did these treatments with their order alternated, the other treatment was broadcasting song from their related population that lives elsewhere, that they do not actually encounter in real life, but broadcasting this song to see if the territory owner would recognize the song and also get angry, also try to kick it out, or whether it would not recognize it as a member of its own population and just ignore the song that we are broadcasting. So we did all these experiments. We did about 13 for each taxon pair on average. And then we calculated a song discrimination metric for each taxon pair as the proportion of territories that totally ignored song from their allopatric relative. And we chose this metric because we consider this to be a pretty clear indication that the birds do not recognize that song as coming from their population when they completely ignore. And we thought these represent instances where song is likely to be a strong reproductive barrier. This metric ranges from 0, where none of the territories that we tested discriminated against allopatric song, to 1, where 100% of the territories that we tested completely ignored allopatric song. So this data set then has 175 taxon pairs for which we measured song discrimination. We also had a measure of genetic distance based off divergence in mitochondrial DNA. So we can place each of these taxon pairs on a plot of song discrimination on the y-axis versus genetic distance on the x. And this is we're basically doing is studying trait evolution. We expect the trait to diverge with greater time. But to bring it back to the central question, we also measured the DR statistic and put, which is an estimate of recent speciation rate estimated from molecular phylogenies, and binned each taxon pair into a quartile of where they fell in their DR statistic. And so the prediction of the reproductive isolation hypothesis is not just that the traits will get different over time, that song discrimination will be greater for taxon pairs that have been evolving independently for a longer time, but that diversification rate quartile will matter. That the taxon pairs that belong to lineages that are speciating rapidly will evolve song discrimination faster. That would be a positive association between the trait evolution and speciation rates, and it would look like this. If, on the other hand, our estimate of speciation rate didn't matter at all, we might see something like this. And one wrinkle is that we did these studies both on sub-Aucine taxon pairs and Aucine taxon pairs. And the difference between these two groups of Passerines is that sub-Aucines have innate song and Aucines learn their songs. And I previously showed that the sub-Aucines actually evolve song discrimination much faster. And the reason why is fascinating, and I'd be happy to talk about that more later. But for our purposes now, it means that we need to take clade identity into account in the model. So we fit a model to predict song discrimination given genetic distance, given clade identity, and then a model that had the diversification rate quartile versus a model that did not have that variable. And this is what we found. What we found is that there is a positive association between how fast the song discrimination evolves and speciation rate as estimated by this DR quartile. And it was present in both sub-Aucines and Aucines, but much more strongly so in the sub-Aucines with innate song. And to kind of help you think about these differences, we can imagine the song discrimination trait value of 0.5, which means half the territories that are tested completely ignore song from the relative, the allopatric relative, and those sub-Aucines with innate song, those in the fastest DR quartile evolve song discrimination to this threshold 2.2 times faster than those in the slowest quartile. A difference of about taking 1 million years to reach that threshold versus 2 million years. And those birds with learned song, the difference is reduced. So what I just showed you is this is one plot talk. I want you to remember that plot. This built up to that plot, and what that plot showed you was a positive association between the evolutionary rates of song discrimination and speciation rates, and it's consistent with this hypothesis that faster evolution of song discrimination leads to faster speciation. There are other possibilities, though, and one among them, kind of a chief thing I was thinking about, was the possibility that there's some other variable that is influencing both of the variables we measured. And of course, we can never completely discount this possibility, but I was particularly interested in the possibility that both the trait evolution and the speciation rate are linked to some geographic variable, in which case the link between the trait and the speciation directly might be spurious. And chief among these is we considered the role of latitude and also elevation, because there's accumulating evidence that recent evolution tends to be fastest at high latitudes, and speciation rates are also often fastest at high latitudes as well. But we found that this was not the case, and I'll show you this with data so that you don't just have to trust me. But when we looked at latitude, here we had to only look at the Aucines, the song learners, because almost all the sub-Aucines are tropical. But there's no difference between the tropical and the temperate Aucines and how fast they evolve song discrimination. Likewise, within the tropics, looking just at humid forest species that live at low, middle, or high elevations, no difference in how fast they evolve song discrimination. Which means that it remains plausible that there might actually be a link between lineages that evolve song discrimination faster, that might kind of directly promote speciation. And so when after clickbaitifying the title, I said, you won't believe these 5 shocking results. So I have to give you 5 shocking results. And the first is that we did find faster song discrimination to be positively associated with speciation rate. And this was a surprise to me, so much so that I'm going to cheat a little bit and put this as the second and third shocking results too, because this really surprised me. Previous efforts to make this link have failed to uncover such a link, and there's many reasons why you might not find this link in nature, like working with real noisy data sets. The fourth result is I'm going to just repeat that we found song discrimination evolves faster in the sub-Auceans with innate song. We had previously reported this. We doubled the data set. We have almost the exact same estimate of the difference between evolutionary rates for these 2 clades. And this is really interesting. It's a downside to learning for speciation. Often we think of learning and cultural evolution and phenotypic plasticity as accelerating trait evolution or accelerating speciation. Here's an example where the trait evolution is accelerated, is faster when the species in question do not have a learned song, do not have that plasticity. And the fifth, again surprising to me, is that latitude and elevation did not affect rates of song discrimination. And this runs counter to some previous studies that have investigated this topic. So I began with a problem. I said there's tremendous variation in how fast lineages produce species, and we're really not very able to explain this. And what I've shown you is some evidence that how fast populations of birds evolve different songs, a reproductive barrier, explains a small part of this variation. And it suggests that rates of reproductive isolation may be important. Clearly, there's lots that remains to be explained, but I think it points to a role for lineage-specific rates of reproductive isolation to be one reason why some lineages produce species more rapidly than others. And so thank you, and I'd love to take any questions you have in these last couple of minutes, or you can reach out and find me on Twitter, which is increasingly used for its core purpose of talking about birds now these days.
Thank you for those shocking results, Ben. That was funny. Yeah, I think we have time for a question. Or Bill?
Yeah. Oh, Nick. Yeah. Nick says, cool results. Could the Aucean result be influenced by the number of co-occurring congeneric or confamilial species?
All right. Okay. Let's see. Let me see if I get this question right, Nick. So could it be influenced by the number of co-occurring species? Like, I don't know. Is the real answer? I don't suspect that it would be because we find the difference when we look even within, say, tropical latitudes for Auceans and sub-Auceans. Like geography doesn't matter, and if it was driven by species richness, you'd think geography would matter.
So I'll ask a question. So what do you think, how do you think the variation in the aggressiveness of birds, you know, they're territorial, but they're not equally territorial. How do you think that might play into this, into how it affects your results and then how it affects your interpretation?
Mm-hmm. Yeah, so the concept about, you know, species are going to vary in their territorial behavior. We only did these experiments on species that really do use song as a territorial defense behavior. And so I think that, and we indexed the song discrimination against the positive control of how they responded to their own local population. So I don't think differences in that kind of starting level would matter. But it does also get at the point that we use a territorial behavior as a metric for a reproductive choice. But I didn't get a chance to include it here, but we do have some evidence that that's a valid proxy, a valid logical thing to do.
Cool. I think it's time for us to move on. Thank you very much. That was great. Our next speaker is Don Miles. Title is Morphological and Performance Consequences of Hybridization Between Marine and Land Iguanas.
Don, you're still muted too.
Okay, how's that?
Good.
Okay. Let's go. I'm pleased to be able to present some research about some studies that we've been conducting on lizards of Galapagos. This is a collaboration between myself, Howard Snell, Heidi Snell, and Paul Stone. So one of the things that got us interested in this is the discovery of a hybrid iguana on Plaza Sur. And we began thinking about what are the evolutionary consequences of hybridization. And so past research considered hybridization to be a deleterious consequence on fitness of the offspring, and this would result in the selection of hybrids, which would reinforce reproductive isolation among incipient species. However, recent data suggests that hybridization may in fact generate new species, often acting through the production of novel phenotypes as a consequence of two species hybridizing. Now, however, there's limited data on the functional consequences of hybridization. And if there are functional differences in hybrids, it may affect how these hybrids exploit resources, evade predators, and it could limit their persistence in current ecological conditions. And a couple of classic examples showing the importance of hybridization in generating new phenological or phenotypic diversity and new species comes from Darwin's finches in Galapagos, and the role of hybridization in the generation of species in the Rift Lake cichlids. Now, one of the things about the study of hybridization is the phenotypic expression of F1 hybrids in nature is often underexplored. And in those studies that have looked at the phenotype of hybrids, they tend to focus on fitness-related traits such as survivorship, fecundity, and growth. And if you look at the particular phenotypic traits that characterize hybrids, there's little consensus on what the expected phenotype should be. Potential patterns could be an intermediate phenotype between the parental species, a mosaic of parental and intermediate characters. Hybrids may resemble one parent over another, or there could be a mismatch between the hybrids and parental species. And a review of this is Thompson et al. In the American Naturalist, coming out in 2021. So one potential outcome is the hybrid has some intermediate phenotype and does not resemble the parental species, or it may be skewed towards one or the other parental species. So the goals of this study was to characterize the phenotypic traits of hybrids and compare them with the parental species. And we focused on functional traits, in particular morphological traits and physiological performance of the hybrids compared to the parental species. And we selected those because both of these traits provide an index into the exploitation of the environment by organisms. And they also can provide an index of resemblance of the hybrid to the niche of the parental species. So this is the star of the show. This is a hybrid marine iguana, land iguana hybridization. So the players are these two species, the marine iguana, Amblyrhynchus cristatus, and the land iguana, Conolophus subcristatus. And one of the things that makes this interesting is that, of course, to have hybridization, you have to have spatial and temporal overlap with respect to the reproductive season. Now, this is a map of the Galapagos archipelago, and you can see there are these yellow and green dots. The yellow dots are land iguanas, the green dots are marine iguanas, and land iguanas and marine iguanas are syntopic on 7 islands. And beginning in 1977, Howard and Heidi Snell began a long-term study on the population of ecology of land iguanas in Galapagos. And in addition, they considered or began an extensive survey of land iguanas on Plaza Sur beginning in 1979. And one of the outcomes of their work was an analysis of the spatial variation in mating and lay dates in land iguanas. And what you see in this graph is in black, you have the mating dates for land iguanas, and then the lay dates, the time that females lay their eggs. Now, there's quite a bit of variation about the archipelago, but I want to point out the mating date and lay dates here on Plaza Sur. So the iguanas begin mating in January, and then the females lay their eggs in April and May. So if you look over on the other panel for the marine iguana, this is looking at the mating behaviors, the number of copulations of land iguanas on Isla Santa Fe, which is close to Plaza Sur. You find that there's a same pattern of mating. And so on Plaza Sur, the lay date of the land iguanas and marine iguanas coincide, which makes hybridization possible. So this is the site where we conducted most of the, well, in fact, all of the work. And in 1981, Howard found a putative hybrid land and marine iguana hybrid. The hybrid was confirmed by Cornelia Rassmann in a paper in 1997 using restriction length, fragment length polymorphisms. Although the identity of the parents, whether there was a male land iguana and a female marine iguana or vice versa, was speculated. We think it's a male land iguana. These are some shots of a land iguana attempting to copulate with a female marine iguana. And there's quite a bit of tussling, but the result is that we do get evidence of hybridization between the two species. So the goal of the study was whether there was evidence of morphological differentiation between the hybrid and the parental species, and whether the hybrid individuals exhibited an intermediate phenotype between land and marine iguanas. The other thing was to look at the physiological and performance consequences of hybridization. And we looked at field active body temperatures, which would give an index of where the animals were active on the island, and then locomotor performance, specifically endurance capacity, because that gives you an idea about the capacity for prolonged activity by the organisms. And we focused on hatchlings because in our survey of hybrids, during the time that we were on the island, the most common age class on the island were hatchlings. And so we were able to get a suitable sample for hybrid hatchlings, marine and land iguana hatchlings. So the data consisted of morphology and performance traits. We had surveys between 1993 and 2000. And in this period of time, we were able to obtain data on 10 hybrid hatchlings, 17 marine iguana hatchlings, and 52 land iguana hatchlings. And so we measured endurance by putting the hatchlings on a treadmill and ran them until exhaustion at a speed of 1/2 kilometer per hour. And the time until they were fatigued was our measure of endurance. And then we measured body mass, snout vent length as an index of size, and then we got measurements of tail length, forelimb, and hind limb length. Now for growth rate, we sampled hybrids between 81 and 2003. So we have data on 41 hybrids and about 300 land iguanas. And we took data from Laurie's paper 1990 on the growth rates of marine iguanas. So this is to show you what the hybrid looks like compared to the parental species. This is the hybrid in the middle, the marine iguana on the left here, and then the land iguana on the right. So they're striking in terms of their expression of color, their size, and their body proportions. So in terms of phenotypic variation, we looked at the differences in the body size of the hatchlings. And what you can see here is that the hybrid hatchlings are significantly smaller than both the land iguana and the marine iguana hatchlings. And so the hybrids were about 10.5 centimeters in length. The marine iguanas are the largest, averaging about 12.5 centimeters. And the land iguanas are intermediate in their size. In terms of their growth rates, we have the hybrids in the yellow circles, the marine iguanas in the green triangles, and then the land iguana in the brown squares. What you can see is that the growth rates of the marine and hybrid are very similar. In fact, the growth parameter K is 0.14 for the hybrid, 0.11 for the marine iguana. So the hybrids are growing as fast, if not faster, than the marine iguanas. The land iguanas have a much lower growth parameter. And in fact, what you see here is that we have data for size up until the 15-year age class. And this is basically as old as we can get in terms of the age of the hatchlings. Laurie was restricted to looking at marine iguanas up until age 6. And land iguanas continue growing well into their 20th year. But the key thing here is that in terms of the growth rates of the iguanas, the hybrids do resemble the marine iguana more so than the land iguana. In terms of their multivariate morphological phenotype, this is a principal components plot portraying the first two PC axes. The first one characterizes variation in size. The second axis is a contrast between body mass against tail length and hind limb length. So basically what we have are animals that are robust with short tails and short limbs versus animals that are slender with longer tails and longer hind limbs. And so that would put the hybrids right here. They're characterized by slightly longer tails and longer hind limbs and a more gracile body than the land iguanas. And then the marine iguanas have a similar sort of pattern, which places them in a more slender or gracile body shape with longer hind limbs and a longer tail. Now, this is shown here. So the land iguanas have a much different shaped tail than the marine iguana up here and the hybrid here. So the marine iguana uses the tail for propulsion when swimming into the ocean for foraging. In terms of the discriminating amongst the groups, we see that the three groups are significantly separated in a morphological space. The marine iguanas, the land iguanas are here, the marine iguanas here, and the hybrids are here. So in the morphological space, it appears as though the hybrids somewhat resemble the land iguanas, but there's very low overlap. And in fact, if you look at the classification or the confusion matrix, it shows that the majority of groups are significantly classified. There's only one misclassification in the hybrids. So it suggests that morphologically, the hybrids are occupying a distinct morphological space compared to the parental species. In terms of physiological differences, we find that the hybrids have a similar body temperature to the land iguanas, and the marine iguanas have a much lower body temperature. This is a reflection of the differences in the habitats that the hybrid and land iguanas occupy. So these are in the more interior parts of the island, whereas the marine iguanas are on the intertidal and are occupying a much cooler environment than the other two groups. What's notable is the differences in stamina between the hybrid and the two parental species. The hybrid has a stunningly high level of endurance. So we recorded the hybrids having an endurance of over 1,500 seconds, which is a long time watching an iguana move along a treadmill. The marine iguanas have the second highest endurance, and the Conalophus hatchlings have among the lowest levels of endurance I've seen in a large lizard. And this reflects a lot of the feeding style of the iguanas. They tend to spend quite a bit of time waiting for apuntia pads to fall off the prickly pear cacti. So they tend to not have the need for active amounts of movement. The hybrids also tend to be more active when they're on the island. The other thing is the difference in the segregation of hybrids from the land and the marine iguanas. The marine iguanas tend to be concentrated along the coastline, given here on Plaza Sur. And these are the capture sites of the land iguana, marine iguana hatchlings. They occur throughout the island in both the vegetated and in the open area of Plaza Sur. We rarely caught them, or rarely, if ever, saw them being active in the intertidal zone. The other thing about the hybrids is that they are more like a land iguana. So here we have a hybrid on a puntia, a tree apuntia, and looking at their scat, we see that they consume quite a bit of apuntia, portulaca, another shrub species, castella, and grasses. Now, one of the things is that the algae, if it is in the diet, will not show up in a gross examination of the scats. But nevertheless, in looking at the diet of the hybrids and their behavior and their habitat occupancy, they behave more like a land iguana. So in terms of the summary of patterns, we find that hybrids are smaller than the parentals, but they have similar growth rates to the marine iguanas. Their multivariate phenotype is unlike the parental species, but they do have a closer affiliation with land iguanas than marine iguanas in the multivariate space. The physiological patterns, their active body temperature resembles more of a land iguana, and yet their stamina exceeds that of either parental species and is, in terms of the parentals, a marine iguana has higher stamina than the land iguana. And then in terms of their overall ecology, they tend to resemble the land iguana. So what we have here is evidence of hybrid vigor, or perhaps hybrid dysfunction, in which the physiological capacity of the hybrid exceeds that of the land and marine iguana. In terms of their phenotype, the hybrids appear to show a mosaic pattern where they resemble the marine iguana in performance, but they're closer to the land iguana in morphology. And this suggests a mismatch in the trait combinations. And so this could be attributable to dominance in opposing directions, which suggests that the hatchlings of the hybrids are kind of in between in their performance attributes and where they are placed in the environmental niche of the land iguana. The other thing seems to be that the hybridization generates novel physiological phenotypes not predicted by morphology. Now, one of the things is that the hybrids do not tend to live very long, and we presume that it's because of this intermediate ecology that the iguanas are not as efficient at feeding on the mainland as the land iguana, and so they tend to have higher mortality later in life. And with that, I'll end, and I'm willing to take any questions if there are any.
Cool, thank you very much.
Yeah, we've got a couple of questions. Oh, yeah. Mark asks, excellent, Don. Is there an estimate for the last common ancestor of the land and marine iguana species?
Yes, it's about 4.5 million years ago. There was a recent paper in the Proceedings of the Royal Society in 2015. And, and one question is, why don't we see other areas or other islands of the Galapagos having hybrids? And in that paper, molecular data suggests that there are hybridization events that may be going on the island of San Cristobal. So we think that there's a divergence in the mating patterns that prohibits more hybridization. Also, it takes a lot of time to find these hybrid hatchlings. And so in areas where there is the potential for overlap, molecular data suggests that they are hybridizing. So that's a good question.
Okay, Kyoko says, very interesting talk. Given that hybridization occurs regularly, do you think there's any sort of selection for or against the hybrids and hybridization itself, even with the higher mortality rate?
Yeah, that's a good question. The problem is we have marked the lizards and they occur at such low numbers that it's difficult to obtain estimates of selection gradients. We have not analyzed survival data. That's something we do have, and it's something that we intend to look at. And that's another very good question.
Jimenez says, fascinating. Maybe I missed this. Do feeding patterns of hybrids vary depending on La Nina or El Nino?
Yeah, that you didn't miss it. I didn't address it. We have some data, and we haven't examined that, but that's an excellent suggestion. Thank you.
Cool. We're at time, but there's a number of other questions here. I know this is the last one. Perhaps if folks want to address the questions in the chat, and I want to thank all of our speakers here for a great session. Yay. And we'll take a 20-minute break, and we'll be back at 5:40 Eastern with the next session. Thanks, everyone. Okay, it's 4:40. I want to welcome everyone back to the second session, the second part of the session on divergence, coexistence, and hybridization. Our first speaker in this part of the session is Meredith Zedelmoyer. The title is Nitrogen Influences Prairie Plant Population Declines and Local Extinction.
Thanks, Becky, and thank you everyone for being here, and to the organizers for putting together this virtual conference. It's been much better than doom scrolling on Twitter. So today I'm going to be telling you all about a portion of my dissertation work, which focused on different causes of population declines in extirpated prairie species. Plant extinctions are increasing across the globe. I'm going to minimize faces, sorry. So this figure shows the number of plant extinction events from 1700 until the present in biodiversity hotspots in red, so in these areas where we would expect lots of diversity, in biodiversity cold spots where we might expect less diversity, and in the combination in black. And what we can see is that from 1700 until about 1820, plant extinctions are generally not really occurring or not recorded. But then with the onset of the Industrial Revolution, plant extinctions across all types of areas increased really rapidly up until the present. Even though we know that a lot of species are at risk, there's still a lot of variability in our projected extinction risks for many species. And so today I'm going to use 4 methods: historical data sets, comparisons of extinct and extant species, and experimental manipulations and demographic data to try to build some more quantitative links between different types of anthropogenic changes and extinction events. So to go through each of these briefly, historical data sets, such as herbarium specimens or old field notebooks, provide documentation of species that have gone extinct in a particular area and in a particular time frame. And in addition, these historical data sets, unlike extinctions that are recorded in the fossil record, the extinctions in these data sets likely reflect declines due to contemporary environmental changes. Additionally, local extinctions. So this means that these species have gone extinct from a particular area. I'll say locally extinct or extirpated kind of interchangeably. But these local extinction risks often reflect global extinction risks. So understanding causes of extinction at the smaller spatial scale can help with conservation. Second, we can use comparisons of these locally extinct species with extant or still present species to identify differences in some of their traits or responses to global change between these taxa that might be less versus more successful in a contemporary landscape. Finally, most studies of extinction risks rely on observational data of species declines in the field, which is definitely a good way to look at extinction risk, but experimental manipulations can provide explicit tests of particular drivers that we really think might matter. And then finally, we can use demographic data to consider different vital rate responses across an organism's life cycle. So I'll be talking about plant extinctions today, so I'll be talking about survival, growth, flowering, and then reproduction. And we can use this data across the life cycle to link community level patterns of species losses to the population level processes that might underlie those losses. And so this framework really fits into sort of a well-known writer strategy of who, what, why, when, and where. So these data sets tell us who went extinct, where, and when. Then we can understand what were their traits by doing these comparisons. And then experiments and demographic data really help us get at why these species disappeared, what were some of the underlying causes. So for my dissertation, I looked at local extinctions in Kalamazoo County, Michigan, which is located in the southwestern corner of the state. And this area has a really rich botanical history. So it was surveyed between 1890 and the 1930s by Clarence and Florence Haynes, who mostly kept these beautiful field notebooks during the Great Depression. And went to all of these sites all over the county and recorded what they saw. And then in 1947, that was published as the first flora of the county. And then in the 1990s, an undergraduate at a local university began this decade-long project of resurveying some of their sites and talking to local botanists. And so we have these two historical records that give us presence-absence data over time. And from these records, we know that generally rare prairie specialist plant species have been disappearing from the county. So I was really interested in figuring out, beyond simply loss of prairie habitat, what might some of the anthropogenic changes that influence these species losses. And so from these records, I pulled 7 pairs of confamiliar species. Each pair is shown in the same colored boxes. And in each pair, I have one species that's locally extinct in Kalamazoo. And because it's locally extinct, we can still source its seeds from nurseries, from prairies elsewhere in the Midwestern US. And then its paired species is still extant in the county. I focused on nitrogen addition and deer herbivory as potential causes of species losses. Because nitrogen addition causes species losses in grasslands. So in this study from a grassland in Minnesota, we see that the proportion of species that were lost increased as there was more nitrogen input into the system. And then in terms of deer herbivory is also known to cause declines in population growth rates. So in this study on Trillium, the researchers examined population growth rates across a gradient of deer herbivory. And we can see that population growth rates are declining. And at about 18% deer herbivory, the population growth rate actually begins, crosses 1 and begins declining towards extinction. Additionally, nitrogen and deer are really likely to matter in Kalamazoo County. So nitrogen deposition in Michigan is a little bit higher than the US average. So this map shows high in red versus lower levels of nitrogen inputs in green. And so Kalamazoo County is about here. So it's in a higher nitrogen input area. Additionally, the Department of Natural Resources has been tracking deer populations, which were at about 1.5 million in the 40s. So that's that first survey by the Haynes. And then a bunch of hunting regulations went into effect. Deer populations exploded and peaked at about 2.2 million in the 90s. So in that, during the time period of that second set of surveys. So I set up a field experiment to test the effects of both nitrogen and deer herbivory in the style of the nutrient network. So I have these plots that are either fenced so that deer cannot get in, or are left open so that the deer can enter and browse. And then within each of those plots, half the plot has nitrogen applied at sort of a mean agricultural level of fertilization, and the other half has no nitrogen. And I'll refer to a control treatment when I'm talking about vital rate responses, and that is no nitrogen and deer allowed into browse, so natural field conditions. So into each of these half plots, I planted 10 individuals of each of those species and monitored the 5,000 seedlings over a three-year period. In terms of vital rate monitoring, these plants were planted as vegetative seedlings. So every year I monitored survival and growth on individuals, as well as whether or not they flowered. And then if they flowered, I counted the number of flowers, collected a fruit, got an estimate of seed production. And then to estimate how many seeds would contribute to the next generation, I did a seed addition experiment where 100 seeds per species were sown into every plot. So I had a species mean estimate under every treatment for germination. And then these germinated plants were monitored to see whether they were going to establish and become vegetative part of the next generation. Okay, so looking at our vital rate results, this is showing survival under the control and the nitrogen treatment. Pink dots are locally extinct species and gray dots are the extant species. And then red will be the extinct species mean and black will be the extant species mean. And that color scheme is going to go the rest of this talk. And what we can see is that the locally extinct and extant species both have about 60% survival under control treatments, and nitrogen decreases the survival of both of these extinction statuses, but that is especially true for these locally extinct plants. In terms of growth, we saw that extinct plants got very little benefit from nitrogen fertilization, which kind of surprised us, whereas the extant species benefited in the way that we would expect when we add fertilizer to the system. So extant species growth benefited a lot more from nitrogen than these extinct species did. Just to show you some species-specific results as well as the herbivory results, this figure shows you the effect size of each of these treatments on survival in extinct species above the dashed line and extant species below it. Dot size is the size of the effect, and then positive is shown in blue and a negative response is shown in orange. And so nitrogen had really consistent effects on survival. It decreased survival across all species. But the effects of herbivory, as well as the combination of the two, were really species specific, and we didn't detect any consistent patterns. And then similarly, in terms of growth, nitrogen generally increased growth, more so for these extant species. The two species that responded negatively are Daphnias, so they're legumes, and we wouldn't necessarily expect them to really like growing in nitrogen-enriched conditions. And again, we saw inconsistent responses to herbivory and the interaction. In terms of reproduction, we found that nitrogen addition increased the number of flowers produced by species sort of across the board, as well as tended to increase the number of seeds produced per plant. And we also saw that extant species, shown in gray, produced about twice as many seeds per fruit as these locally extinct species. And so this difference in seed production is this general demographic difference between these potentially more successful versus less successful, or at least declining species in this landscape. To summarize the responses to nitrogen that we saw across the life cycle, we saw that survival decreases, especially in locally extinct species. Growth is increased, especially in the extant species. And nitrogen generally increases flower and seed production, so it's beneficial for reproduction. And then there were no differences between nitrogen or herbivory in terms of germination and establishment. And so I think this really points out that we need to be looking at plant responses across the life cycle, because if we had just looked at growth or biomass, we would have assumed that nitrogen was having a benefit for each of these species, and that's not necessarily true. So I then built integral projection models for 5 of the species that had sufficient vital rate data across their life cycles. And so I'll be showing you population growth rates under each treatment, with the control shown in gray. And because this was an experimental setup, so we transplanted these seedlings into a fairly old restored prairie where forbs are not super common. We're not particularly interested in the absolute values of these lambda estimates so much as the relative difference between a given treatment and the natural field conditions. So you'll see these downwards arrows when population growth rates are depressed, and upwards arrows when population growth rates were higher than the control. The other thing I want to note is that for these IPMs, the blue bars are the effect of the fencing. So if there's a positive effect of deer absence, you can think of that as a negative effect of deer herbivory. So first, we saw that nitrogen decreases population growth rates in all of the species that we conducted IPMs for. And on average across species, it decreased lambda by about 65%. So nitrogen was really having a negative effect on all of these plants, regardless of whether they were extirpated or not. In terms of deer herbivory, we found a positive effect of fencing or a negative effect of herbivory in 3 of the 5 species, although the trends were not significant when we looked across population growth rates across these species. I also want to note that the interaction, so when we had both nitrogen added and fencing up, the 3 penstemon species all experienced lower population growth rates. And these 3 species are generally shorter, a little bit slower growing than these 2 mints. Oops. And so when we have nitrogen and deer absent, the vegetation got really, really dense. So I think this effect is mostly due to shading and light competition. So with this data, I was able to do a life table response experiment to figure out which vital rates were most important to those differences in population growth rates. So you'll see the effect of a given vital rate for a given species. And then if it's a positive effect, then it increased population growth rates relative to natural conditions. And if it's a negative effect, then it's decreasing population growth rates. And so the LTRE showed us that negative contributions of survival and growth for both nitrogen in red and deer herbivory in blue were what was driving those lower population growth rates. And reproduction had very minimal effects, which is what we would expect for these perennial species. So in conclusion, if we think about who, what, where, when, and why species went extinct, we see that rare prairie specialists went extinct in Kalamazoo County from the 1890s until the early 2000s. Extant or more successful species produced more seeds per fruit than extirpated species. And it seems like nitrogen addition, so in the case of this particular area, that'll be mostly from runoff from agriculture. These prairies have been pretty much run over for farmland, but specifically lower survival under that nitrogen addition. Ongoing work in this system, we planted these species into this agricultural, pretty high level of fertilization, but they went extinct over the past 100-ish years, where nitrogen levels could have been lower. So we've set up a nitrogen gradient experiment with the same species to look at how that gradient affects their demographic rates, as well as collecting a lot of different trait data to look at the role of species traits, as well as interspecific variation in traits in demography. And with that, I'd like to thank everyone who helped with these projects, especially a lot of undergraduates who were able to develop their own projects within this setup. And I would love to take any questions. Thank you.
Thank you. I think we have some time for a couple of questions, Bill.
Okay, Ellen says that it's very cool research theme, and then asks, it says, you mentioned Baptisia not responding positively to added nitrogen. What did you see from other legumes?
That was the only legume pair that we had in the species. And unfortunately, we don't have great data on it because it pretty much got wiped out in the first year of the experiment.
We have a question from Abdel. Great talk. Are these plant species known to form nitrogen-based microbial mutualisms?
So the legumes, yes, they have rhizobia nitrogen-fixing bacteria. I don't know if the other ones do or not.
So is there any indication that the loss of pollinators might have had some impact over time with some of these extinctions, or the reduction in pollinators?
We don't know. So we've certainly, like the ones that are common in the experiments, like the penstemons, like bees love them. But we have another setup with these species under climate warming. And we're trying to, like, one of our goals for the summer, if we can do field work, is to look at whether the extirpated species have less pollination, either because they're less attractive or because maybe they had specialist pollinators that disappeared sort of concurrently.
Great. Thank you very much. That was great. Our next speaker is Lucas Eigentler, and this is a recorded talk. The title is Species Coexistence and Resource Limited Patterned Ecosystems is Facilitated by the Interplay of Spatial Self-Organization and Interspecific Competition. I'm going to stop my video. There's share. This.
Hello, everyone. First of all, I would like to start with an apology for not being here live today and only sending this talk through this recording. This also means that I won't be able to answer any questions after this talk. But if you do have any questions, I would like to very much encourage you to send me an email. Here's my email address, which will be also displayed throughout the talk in the bottom left corner, or get in touch through Slack. So to get started with this talk, the title is rather long-winded. It says that species coexistence in resource-limited patterned ecosystems is facilitated by the interplay of spatial self-organization and intraspecific competition. So, but I will hope to be able to disentangle this talk title throughout the talk. So to get started, this talk is about patterned ecosystems. And here you can see 3 photographs. The top left is a semi-arid vegetation patterns, which occurs in many semi-arid regions across the globe. The second photo is a intertidal mussel bed taken from the Wadden Sea close to the Netherlands. And finally, these stripes you can see here are usually referred to as ribbon forests, and this is taken from the Rocky Mountains. Now, these patterns occur, these photos, these ecosystems obviously occur in different regions of the planet. However, they have certain things in common. And first of all, usually these patterns occur if environmental stress is high, for example, a lack of water in the semi-arid vegetation patterns. The second thing they have in common is that typically there is a self-organization principle at play that causes this formation into spatial patterns. And these self-organization principles are usually a scale-dependent feedback that consists of long-range competition. So that is competition for the limiting resource. That's why environmental stress plays an important role in these ecosystems, and a mechanism that causes local facilitation, where species facilitate, where individuals facilitate each other if they're nearby. In the case of vegetation patterns, this is a modification of the soil by plants that increase the permeability of the soil, and therefore water can infiltrate the soil much better in vegetated areas. This causes a redistribution of the resource towards areas of high biomass, and this closes the feedback loop. And similar mechanisms of facilitation occur in the other ecosystems as well. Now, these patterns can occur in many different shapes and forms: gap patterns, labyrinth patterns, spot patterns. But in this talk, I want to focus on one particular kind of pattern, which which are these stripe patterns, as suggested here in these photos. And these stripe patterns usually occur if there is a unidirectional resource flux in the system. For example, a slope in the terrain that causes water to flow downhill, or the tide in mussel beds. Of course, the tide goes in two directions, but along the same axis. And if there is such a unidirectional resource flux, then patterns typically occur perpendicular to the flux of the resource. And another very interesting observation of these patterns is that if there is such a resource flux, these stripes slowly move in the direction opposite to the resource flux over time. And this is something that has been reported from both vegetation patterns and ribbon forests, not from mussel beds, because they're usually destroyed during winter storms and need to reassemble. So it's difficult to make any comparisons over time. Now, the talk is also about coexistence, and that is because typically coexistence in these ecosystems is observed on the scale of a single stripe. So this is not about coexistence through spatial segregation, but really coexistence as a single point in space. And coexistence in these ecosystems occurs despite the high environmental stress and despite the competition for just one limiting resource. And I think we all have the principle of competitive exclusion in mind. And so the question I want to address here is, what mechanisms cause coexistence, enable coexistence in these ecosystems? There are two very strong candidates here, one being a very classical result, and that is intraspecific competition. But more recently, it has also been shown that spatial self-organization can enable coexistence. And so in these ecosystems, both these processes seem to play a very important role. And so the question is, what impact does the interplay between these two processes have on the coexistence mechanism? And that is particularly interesting because these two processes, intraspecific competition and spatial self-organization, have quite an opposite effect, because intraspecific competition, as the name suggests, is associated with competition, while spatial organization is typically associated with local facilitation. And that's why the question about what impact the interplay between these processes has is quite interesting. Now, to address this, I have used a theoretical framework, which you can see here. I don't want to go into the details here to keep this talk fairly short, but I would like to point out that, first of all, this model is an extension of an existing single species model that's usually referred to as the Klaus-Meyer model, which is a model that was proposed to explain pattern formation in dryland vegetation patterns. But I actually argue that because the model is so simple and phenomenological, it can also be applied to other pattern ecosystems as well. There are two species, U1 and U2, that interact with a limiting resource called W here. The processes here are I don't want to go into the details here, but I would like to point out that species U1 and U2, if you look at these two equations, you will observe that they don't differ qualitatively. So the functional responses are the same, and they only differ quantitatively through the parameter values. And so in particular, U1, I have just assumed this U1 is a species that is superior in its colonization abilities, whereas U2 is a species that is superior locally. And I would explain in a second that this is an important ingredient in the coexistence mechanism. Now in this model, intraspecific competition is only accounted for indirectly through depletion of the resource, through the interaction with the resource W. However, other intraspecific competition dynamics may also play an important role in these ecosystems. And so I further extended this model through the inclusion of these logistic type terms, which simply say that an increase in the abundance of a species limit its growth. And so through this, I will be able to also get an insight into how intraspecific competition dynamics, other than that for the limiting resource, affect species coexistence. But to start with, as a baseline case, I will show you results for the model without these additional intraspecific competition dynamics. So having shown you the model, the first question, of course, is does this model actually capture coexistence? And the answer, of course, is yes. Here you can see just one snapshot from a numerical simulation of the model. On the top, you can see both consumer species densities plotted on top of each other, and you can see that they happily coexist. However, you also see that in this particular solution, the solution actually does not represent a patterned ecosystem, because the solution oscillates between two non-zero values. Whereas in a truly patterned, in a solution that represents a patterned ecosystem, you would expect a solution that oscillates between a high value and zero. And this is not the case here. And I will point out later that the reason why this is not the case here is that for this particular solution, the environmental stress was not set to be high. And in particular, to capture species coexistence under high environmental stress, intraspecific competition dynamics need to be considered. And so the suggestion is that this plays an important role. However, this observation is still very significant because it shows you that species coexistence is enabled by just the spatial self-organization principle. And that is because this induces also heterogeneities in the resource, which, if there is a balance between species colonization abilities and their local average fitness, the superior colonizer can utilize these heterogeneities by colonizing new areas before eventually being outcompeted by the locally superior species. However, at the same time, the colonizer species already colonizes new areas. And so this creates a balance that enables species coexistence. Now, one thing you can't see here, but which is very important, is that the model actually also captures the movement of the patterns in the direction opposite to the unidirectional resource flux. Now, this is just one snapshot in time. However, I've made this more detailed, the results, by really looking at mapping out the stability regions of the different pattern solutions in the whole parameter space. And here I'm using a two-dimensional parameter space. On the x-axis, you have the main bifurcation parameter of the system, which is the resource input parameter. However, for every single set of parameter values, we typically observe many different pattern solutions that occur at different wavelengths, and in particular also different migration speeds. And therefore, the y-axis here is the migration speed. Now, what you can see here is the two stability regions of the coexistence patterns in yellow and the pattern of the colonizer species, where we just have the colonizer species in blue. Note that the pattern, the single species pattern of the locally superior species, is never stable. And this is for a case with no interspecific competition dynamics or weak interspecific competition dynamics. And the main thing for you to take away here is that here, that point, where the transition from coexistence to single species occurs, so where an extinction event occurs, this happens at relatively high levels of the resource input parameter. And that is the reason why, in the absence of intraspecific competition dynamics, the model only captures coexistence if environmental stress is not severe. And so this brings me to the role of the additional intraspecific competition dynamics, because if you include those, the picture changes. And in particular, if intraspecific competition among the colonizer species is sufficiently strong, the transition where the coexistence state loses its stability to the single species state of the colonizer species shifts to lower values of the resource input parameter. And I will show you an example solutions later on, but in particular here in this parameter region, the solutions in this case really represent a patterned ecosystem where the solution oscillates between a high level and 0. And so the main message to take away from here is that by including intraspecific competition dynamics among the colonizer species, the model actually captures coexistence in a patterned state also. And so the suggestion is that while the coexistence mechanism is the spatial self-organization principle, intraspecific competition dynamics also play an important role under high environmental stress and are required under high environmental stress to enable coexistence. Now, the other important thing here I would like to point out is that intraspecific competition dynamics also have an impact on the single species pattern state. If you look at where the single species pattern state loses its stability, here in the absence of intraspecific competition dynamics, you see that by neglecting intraspecific competition dynamics, you overestimate the resilience of the patterns to environmental stress. And so this again highlights the importance of intraspecific competition dynamics in modeling of these ecosystems. The other thing I would like to point out is the importance of hysteresis or history dependence. Now, this is a slightly different picture. Here, the intraspecific competition among both species is strong, but this doesn't really affect the transition between coexistence and single species states. I've also superimposed wavelength contours here of 150 and 25. And the thing I would like to point out here is that the transition where an extinction event occurs cannot be pinned down to one single parameter of the resource input parameter. And so in particular, the value where transition occurs depends on the wavelength of the solution. So if you start off here at a solution with a wavelength of 100, and you slowly decrease the rainfall parameter, the resource input parameter, I should say, then the transition occurs here where the locally superior species just goes extinct and the solution continues along that wavelength contour in a single species state. However, if you were to start at a pattern of a wavelength 50 at the same resource input parameter, you could decrease the parameter to lower levels without losing the locally superior species. And so this suggests that hysteresis plays an important role. And similarly, you can also increase the resource input parameter here. And as soon as you hit a stability boundary, which happens here, you swap to a different solution at a different wavelength. And you can see here you jump from 100 to 25 rather than going to 50. And this is actually a very interesting phenomenon, which we currently don't have a handle on which new wavelength is selected. And this is definitely something to look at in the future. And finally, I promised to show you an example solution. And here you can see actually two. We really see that here there is an oscillation between high levels and 0. So these solutions really represent a patterned ecosystem. The other thing I would like to point out here is that the model captures something that has been reported from vegetation patterns, which is that one species is confined to the center of each single stripe. And we have through numerical continuation, I've shown that this really is due to the dispersal behavior of the species. So faster dispersal means that the species is located more to the edges of each single stripe. So just to conclude, I have shown you through this very simple phenomenological mathematical model that coexistence in patterned ecosystem is enabled through a spatial self-organization principle that induces heterogeneities in the resource, which a species that is superior in its colonization abilities can utilize before it's being outcompeted locally by a locally superior species. This creates a balance that enables coexistence. However, it only manages to enable coexistence if environmental stress is not high. And to really capture coexistence in patterned ecosystem, where environmental stress is high, intraspecific competition dynamics need to be considered. There are other coexistence mechanisms as well in the system, which I haven't had time to talk about, but coexistence can also occur as a metastable state, which is a long transient state that occurs if local average fitness differences between species is low. And so just to finish, I believe that this is a very rich topic and there are many open questions that still need to be addressed. Here, I've written down some of them. I do have answers to those questions, what the impact of those questions is in terms of single species models, but I believe that all these different mechanisms may also have an important role to play in species coexistence. And so there are many avenues for future work here to be explored. And so just to finish, I would like to point out that the main content of this talk can be found in this preprint here on bioRxiv. But if you're interested in some more mathematical details on how these results were obtained, I would also like to point you to these other references as well. And with that, I would like to finish and thank you for listening. Thank you.
Excellent. So it is, I think we're almost ready for the next speaker. I guess the thing I'd say is if you have comments or questions or want to get in touch with that author, please just let me know and I can drop his email in the chat. So our next speaker, yeah, we're right on time, is Freya Rowland. The title of her talk is Synchrony, Density, Dependence, and Persistence in Amphibian Populations. And Freya, I can't hear you.
Yeah, I forgot to unmute myself before I started, so let me try this again. Okay. Hello, everyone. I appreciate everyone coming at the very end of this excellent conference to hear me talk about some of the work I've done for my postdoc with Dave Skelly. Today, I'm going to talk about the wood frog, Rana sylvatica, which is useful as a system to study population ecology for a number of reasons. One, it's got a really broad range, anywhere from the deep south all the way up to the Arctic Circle. The lifespan of the wood frog is about 2 to 5 years, making it fairly tractable for studying multiple generations. Males become sexually mature at 1 year and females at 2 years. Like most pond-breeding amphibians, they've got really high breeding site philopatry, which means that they come back to the same pond to breed that they spent their larval stage in. And if they do move, in the rare instances where they do disperse to a new population, that move happens during their juvenile stage, right as they metamorphose. And so we can follow populations within a pond pretty well. And then also very useful is that each female only lays 1 egg mass per breeding season. And so what I'm going to talk about today is this question about what drives population trends in wood frogs. A lot of our ability to understand drivers of population trends is hampered by the fact that we don't have a lot of long-term data. But that is not the case in Dave's lab. When Dave started his his lab at Yale, he started a long-term research project at Yale Myers Forest, which is in northeastern Connecticut, where for the past 21 years, the lab has followed 64 different populations and 64 different ponds across all of those years. And so Yale Myers Forest is a managed forest, but it's not had a lot of land use change, where they do log it, but it's still relatively perfect wood frog habitat. Also quite useful with wood frogs is that they tend to lay their egg masses in these big egg mass rafts. So they're like many amphibians, they're an explosive synchronous breeder in the spring. And the whole forest, all of the ponds across Yale Myers Forest will tend to breed within 2 weeks. And so we can go to the ponds and it's pretty easy to tell whether they're bred or not, because you'll find 1 to 3 of these egg mass rafts. And this one in this picture is particularly large. But each one of these lumps is a different egg mass that represents one female's breeding effort for the year. And these 64 ponds also are quite different in their habitat. So they vary in size from very small to very large, canopy closure from pretty open to relatively closed. These are all vernal pools for the most part, and so they're mostly pretty shallow, about 0.5 meter deep. But the populations in these ponds are quite different. So we can have ponds that have no egg masses or 1 egg mass per year, all the way up to ponds where we count more than 1,000 egg masses within 1 year, which is remarkable. And so kind of returning to this question, what drives population trends in wood frogs? Due to a lot of really excellent population community ecology, we know that the pond matters. We also know, based on these studies that have looked across broad regions, that regional level variation matters. You know, climate matters for determining what's going on in populations. But really comparing these and find enough data to get some good estimates of how different pond level and regional level factors influence population dynamics, I don't think it's been done. So for pond level factors, today I'm going to show you some data on canopy cover. Hydroperiod is just how long a pond holds water. These are complex life cycle species, so they spend their larval stage in the pond, and then they metamorphose and spend their juvenile and adult stages on land. And so they need to have enough water to make it through a season and metamorphose, otherwise there's complete cohort failure. Also, intraspecific competition. A lot of these ponds, wood frogs are the main player. There are other species that are there, but in terms of population size, wood frogs probably are mostly competing with each other. And then depth, which also relates to hydroperiod. On the regional level, and here I'm talking about across the forest as my region, I really want to know whether there's population synchrony. So are these populations rising and falling together, or are they independent of each other? How do wet versus dry years influence the populations? Terrestrial competition, once they leave the pond, we don't know much about what they do on land. And then how are winter temperatures affecting population trends? I'm going to show you the data assessed 2 ways. So one is just looking at the population size. We can go and count the egg masses, and it gives us a very good proxy for how many females are in the population, at least reproductive females. But we can also look at population growth rate. Because they have a relatively short lifespan, and because females are only reproductively viable at 2 years, if we look at the log ratio of the count of egg masses in the current year relative to the count 2 years prior, it gives us a pretty good proxy for how those individuals that were in egg 2 years previous, how many of them are coming back to breed. And so this is a proxy for population growth rate. So here I've plotted all the different 64 ponds across time. I do not want you to focus too much on this. But what I wanted to highlight was that we've got a lot of data, we've got a lot of variability in the data, and we've captured a lot of different types of populations. So on the y-axis is the year from 2000 to 2020. And on the x-axis, rather, on the y-axis is our wood frog egg mass count, and why my y-axes are different. But I want to highlight that we've got the gamut of different types of populations. We've got some ponds that are increasing in population size. We've got some, like Little Thing, that were extirpated, where when they started monitoring, the population was fine, and then it just decreased to local extinction. Some ponds like X7 collapsed and then have rebounded again. And then we have some ponds that appear to be declining over time. And so I'm going to show you the data split by this pond reliability, which is just the proportion of years that a pond had egg masses during surveys. And so all the figures I'm going to show you are split similarly. I'm going to show you some Bayesian hierarchical models for the data, but I just want to show what the ponds look like if we look across all the years. So we had 23 ponds that always had egg masses every year that we went to them. And we had 24 ponds that had egg masses the majority of the time, about 75% of the time on average, but less than 100% of the time. And then we had some low reliability ponds, 13 ponds that had egg masses less than 50% of the time. I also want to highlight that even though these always reliable ponds always had egg masses, that doesn't mean that they had high population sizes. So we had some ponds that on average had more than 250 egg masses per pond per year. And then we had some that were close to 0. So first, I'm going to talk about the pond level factors that are affecting these population dynamics. So this will include canopy cover, the depth of the pond, and then intraspecific competition in the larval stage. So the figures I'm going to show you going forward are all betas for regressions between the parameter of interest, so in this case, depth, and then egg mass counts will be on the left column, and population growth rate will be on the right column. And so deeper ponds tend to be better. This makes sense because a longer hydro period means that you're going to have more metamorphs coming out of that pond. We can kind of see this in growth rate, it's not supported, but there's not really any discernible trend with canopy. These canopy values are all over the place. And these points are the mean, and then this is the credible interval around it, the 50% and 90%. So we don't see any trend with canopy, which is not all that surprising considering that wood frogs are generalist. Also, we found that density dependence is highly likely. So here we've got density 2 years prior, how that's affecting individuals coming back to breed. So if you have a high density 2 years prior, you're going to have more egg mass counts. So if there are a lot of individuals in a pond, you're probably going to have more returning. But just because a pond has egg masses doesn't mean that it's doing that well in terms of population growth. We can see a strong negative effect of density 2 years prior on the population growth rate. And then I just had this tadpole pop up to remind you that this is interspecific competition in the pond, in the tadpole stage. We can also look at the effect of neighboring ponds on a focal pond. And so if I look at the effect of ponds within 500 meters of the focal pond and how many eggs they had 2 years ago on a focal pond, we see a positive effect for egg mass counts, but in general, there's a hint of a negative effect for population growth rate. So this could indicate that there's some sort of terrestrial competition or density dependence in the terrestrial phase. Switching gears a bit, let's look at regional factors. So we're going to look at synchrony, winter thaw, and then the Palmer Drought Severity Index, which is an indicator of the moisture in the soil, really. I'm way oversimplifying it, but if it's negative, that means that it's a drought condition. Positive means that there's excess water on the landscape. And we can also look at that 2 years ago to see if conditions in the climate 2 years ago affected the present. Surprising to me at first was that there was no synchrony in this data at all. These are synchronous breeders, so I thought that maybe the populations might synchronize, but they do not. And we also didn't find any spatial autocorrelation. There's no relation between how close ponds were together and the egg mass counts, with the exception of 2016, which was just a random year as far as I can tell. I've done multiple different analyses, including kriging, and we always get the same result. Thinking about winter, what's interesting is that wood frogs, and I have this picture here of this frogsicle, are one of very few species that have a compound in their body that's similar to antifreeze. And so they can freeze solid and then thaw and continue to survive. And so I used this metric called winter thaw, looking at the number of days during the winter where the temperature was above freezing. And so the more days above freezing gives a really strong negative effect on egg mass counts and also on population growth rate in these highly reliable ponds. And so I don't know if this is because of direct mortality or whether they didn't have enough energy for breeding effectively after that, but that's something that definitely deserves further research. And then looking at PDSI, this Palmer Drought Severity Index, wetter years are better for egg mass counts, maybe for growth rates. And then we don't see a lag effect as far as I can tell, where a wetter year 2 years ago in the present time results in higher egg mass counts, but no real effect on population growth rates. And I had this pop up because I want to make the point that the moisture in the landscape matters not only just for the larval stage, but because they have this complex life cycle, they need to have enough water to make it through their larval stage. If the pond dries before they can metamorphose, complete cohort failure. But also when they leave the pond, they are very susceptible to desiccation. And so I can't separate those two effects out, but I think that there's some interesting suggestion that this moisture in the landscape really matters, not only for just how many ponds are available, but also for what happens once they metamorphose. So I've shown you a lot of data. Let me try to summarize it in a way that's a bit more tractable. So we have these pond level factors, including the density in the pond 2 years prior, pond depth and canopy, and then these regional level or across the forest factors, winter thaw, and PDSI 2 years prior. So we saw that in general, there are a lot of these things had a positive effect on egg mass counts. We have density 2 years prior means higher egg mass counts. Pond depth has a positive effect. More moisture is good, but the strong negative effect of winter thaw. Oops. Uh-oh. But at the population level, I don't know if it's not popping up. At the population level, this effect of density was actually negative. And so just because there are egg masses doesn't mean that the population is doing well, although most of everything else was pretty similar. So what are the main drivers of wood frog population dynamics? We've got some good evidence that intraspecific density within the pond matters, and that matches a lot of previous literature. Depth and Palmer Drought Severity Index matter. And I think that this matters a lot because of, like I mentioned, not only having a pond to breed in and having the pond be wet long enough to make it through metamorphosis, but also because amphibians tend to lose water very rapidly. And so having more water is beneficial once they metamorphose. And then the strong negative effect of winter thaw, where the more days above freezing, the worse the frogs seem to do when it comes to the breeding season. And what's interesting is that in Connecticut, the number of days above freezing has actually been increasing, but the mean temperature is not changing that much. And so there's definitely some climate change implications. And also for me, I consider myself an aquatic ecologist. These data showed me that it's time to address my aquatic bias. The pond is important, there's no doubt about that. But what's going on in the terrestrial habitat really matters too. We saw some evidence of terrestrial competition, that drought matters, and this winter temperature effect. So to wrap it all up, normally when we think about managing populations or thinking about establishing a study site, people tend to focus on a pond that's doing really well. But focusing on one pond misses all the ponds within that landscape. And having more ponds that you try to manage or conserve can be really beneficial. This asynchrony in the population is great because if there's a catastrophic event that wipes out one of these ponds, like this pond, we can have a rescue effect from the other populations around it. But also ponds exist in this terrestrial heterogeneity too. Like there are trees, there are rows, there are all sorts of things that you need to think about. And then there are these top-down forces like temperature and precipitation that also affect what's happening. I do want to note that there were no big differences between the pond reliability groupings, which is great because a lot of people don't follow the ponds that don't have high populations. And so it's cool that regardless of whether it's a pond that has a really robust population where they're always there, or just a population that comes and goes, that we found similar effects of these different pond level and regional factors. I also want to thank the many, many people in the lab who contributed to this. I definitely was not here for 21 years collecting these data, and my funding sources, and my postdoc advisor, Dave. And with that, I will open it up to questions.
Great, thanks. Yeah, there's time for some questions.
Yeah, a question from Becky. Are the little frogs coming out of the pond too soon when there are too many days above freezing?
So I probably should have mentioned this in the life history. So they breed in the early spring and they metamorphose in early to midsummer. And so it wouldn't be a problem for them metamorphosing in the spring. It'd be maybe breeding and having egg masses freeze. That could happen and does happen.
Just to follow up with that. So temperature affects two things. It can affect growth rate and development. So if you have more warmer days, you might accelerate development, but not necessarily growth rate, depending on the resource conditions. So that I think that, conceivably they could come out a little less viable as metamorphs if they've undergone faster development. From Mark, excellent talk. Goldenrod galling taphritid flies die at higher rates in warmer winters because they use up their winter energy stores faster. Do you think the same may be happening with overwintering in the wood frogs? Has anyone looked at energy storage and metabolism in the winter in wood frog adults?
Excellent question, and not that I know of. There is a new PhD student in the lab who wants to look at what happens with this winter freezing and thawing and how that affects the metabolism of the frogs, but I don't think there's a lot of information about it right now.
Maddie has a question. You mentioned deeper ponds have more egg masses. However, deeper ponds, if they are permanent rather than ephemeral, have more tadpole predators. At least we see this in New Hampshire. Could more egg masses in deeper ponds be a result of females compensating for higher predation rather than because they are better habitat?
The large majority of these ponds are ephemeral. And so we don't have any ponds that have fishes, which is why I didn't include any predator effects. But for sure, there is an effect of salamanders and also dragonfly larvae. I think that in this case, we aren't talking about huge differences in depth. I mean, we're talking about 10 centimeters difference. And I think that it has to do more so with the hydro period. But that is a good point, that ponds that keep water for longer tend to have more predators.
So from Volker, what about phenology? Do you see differences across pond categories, years, etc.?
So Andreas, who's a PhD student in the lab, just published a paper where he found that they were not, I believe, but we haven't explored that in terms of this pond reliability grouping. So that'd be a good idea to do.
I have a quick question. So I always struck by a little bit of tension between the idea of rescue effects and philopatry. Can you comment on that? I mean, if you're really highly philopatric, how do you find that other pond, or how does that other pond draw you to it in a bad year?
Yeah, I mean, I think that that's definitely helped by the fact that these are a calling species. I worked with salamanders during my PhD, and they definitely did not call. But I think that might be a big effect there. I know that the lab is looking at a lot of genetic data to try to figure out what the dispersal rates are. I mean, the data we have on dispersal and saying that there's really high philopatry typically come from a handful of ponds. It's really intensive to try to collect those data. And so this 10% dispersal rate, we don't really know what it is. And so, I mean, it's definitely something we need to explore more.
Okay, thank you very much.
That's great.
Our next speaker is Jen Coughlin, and the title of the talk is the genetic architecture and evolution of life history divergence among perennials in the Mimulus guttatus species complex. Take it away.
All right, everybody hear me okay?
Yep.
Good, and everyone can see the slides. All right, I'm gonna hide your pretty faces for the moment. Oh no. I'm trying to do this newfangled thing where you just show part of what I can see. Okay, let's give that a try. All right, my name is Jen. I'm currently a postdoc at the University of North Carolina. And today I'm going to talk to you about a project that I started in my PhD, but I've been working on for the last couple years. And it's about the evolution of life history divergence in perennials. Okay, So I'm sure I don't need to convince anybody in this room that ecological divergence both shapes and is shaped by trait divergence between closely related species. And this largely comes about through the phenomenon of local adaptation, which is one of the most ubiquitous phenomena in evolutionary biology. Local adaptation has a lot of implications for species coexistence, at least at broader geographic scales, and also in terms of reproductive isolation between incipient species or more divergent species. Today I'm going to focus on plants, and I'm specifically going to focus on one of the main targets of local adaptation in plants, which is life history divergence. And one of the most, in my opinion, classic examples of life history divergence and local adaptation is in these two different ecotypes of Mimulus guttatus. So Mimulus guttatus occurs as these two forms. There's a perennial form. You can see it's highly vegetative, and it produces these vegetative stolons or runners. You can see them dangling off the pot there in this photo. This is in very stark contrast to an annual version, where annuals have this sort of live fast, die young life cycle. They invest a lot in a very quick reproductive output, and they don't have very much vegetative biomass. These species live in really different habitats, or incipient species live in really different habitats, which is perhaps unsurprising. So annuals live in these ephemerally wet conditions that suffer the seasonal onset of drought, while perennials live in these consistently wet environments. And life history in this system confers really strong reproductive isolation by this reciprocal local adaptation. But I want to just pause here for a moment and say that life history is more than just a single trait. It's in fact a suite of traits. And in fact, actually, we can think about it as an investment strategy of a plant. And so if we're thinking about investment strategies, we might think about how these strategies change across time, across development. And particularly if we're interested in divergence, we might want to think about how development changes across time. Okay, the second sort of important thing to remember is that if we're interested in how local adaptation shapes reproductive isolation, it's important to think about the genetic architecture of local adaptation, and this is for two reasons. First, the genetic architecture of trait divergence strongly influences how easily that trait divergence can evolve to begin with. So for example, strong trait correlations can either facilitate or constrain trait evolution. And on the other side of this, the genetic architecture of trait divergence influences how traits are expressed in hybrids, which of course influences their fitness in nature. So today I'm going to try to address these sort of concepts, again using Mimulus, this case using the whole Mimulus guttatus species complex. And this is because the Mimulus guttatus species complex is this hugely diverse group of species. And we know a lot about life history, predominantly through these really classic and beautiful studies contrasting annual and perennial life forms, but also a lot of work that's gone into studying annuals in the species. But today I'm going to focus on the pink elephant in the room, which are these perennials. So the Mimulus guttatus species complex has this suite of perennial species that are much less studied. And today I'm going to talk about 5 of them. So inland perennial Mimulus tillingi is this high elevation subalpine plant. We have 2 different forms of Mimulus guttatus. There's a coastal perennial form and an inland perennial form. And then there are these 2 perennial endemics, Mimulus corallinus, which lives in the Sierras, and Mimulus decoris, which lives in the Cascades. And today I'm going to ask, do these perennials vary in their ecology? Are there any life history differences between them, and are these related to differences in ecology? And lastly, what's the genetic architecture of life history divergence? So to answer this first question, I first constructed these cross-validated maxent models for each taxa using herbarium records. I then predicted suitable habitats and the predicted range of each species and randomly sampled within these ranges to get a distribution of a lot of different ecological variables, including elevation, which is going to be important for this talk. So the results of that are shown here. On the left, what I'm showing you are the predicted distributions for all 5 taxa. And really what I want you to take home here is that these taxa vary tremendously in how geographically widespread they are. So inland perennial Guttatus has this very widespread distribution, whereas Mimulus coralina, shown in this little orange dot here, is really largely restricted to the Sierras. And these differences in distribution also translate to differences in their elevational niche. So we have 3 species that are largely restricted, or at least largely occur at these mid to high elevation sites. So Mimulus coralinus, Mimulus decorus, and Mimulus swingii. In contrast, inland perennial Guttatus has this really wide elevational distribution, and coastal perennial Guttatus, as the name might suggest, lives along the coast, which are really low elevation habitats. So to assess how these perennials vary in life history, I also completed a common garden experiment with about 200 plants from 40 populations. And then I surveyed a suite of vegetative and reproductive traits at 3 different developmental stages. Before the plants flowered, so a vegetative stage, on the day of first flower, and then several weeks after the plants flowered. And what I'm going to show you are these suites of traits as portrayed by a principal component. So in this case, only the first component was significant across all of these traits. And values of PC1, greater values of PC1 indicate a greater investment in vegetative biomass. So you can think of these as like the super perennial plants. So we can see early in development that these species don't have a lot of differences between them. But as development proceeds, we start to see some differences emerge. And in fact, late in development, what we find is that these 2 endemic species, Mimulus coralinus and Mimulus decorus, invest a lot in vegetative biomass and have delayed reproduction, whereas coastal perennial Guttatus and inland Guttatus, much less so. We can also contrast these investment strategies in the context of where these populations originally came from. And so here what I'm showing you is that value of PC1, but now just regress across the elevation of origin for these populations. And what we find is that there's a relationship here such that plants from higher elevations invest more in vegetative biomass, but we really only see these relationships much later in development. Now, what was really interesting about this phenotypic survey is that in addition to these differences in sort of multivariate trait space, we also see the production of a unique trait, at least unique for this species group, which is the production of these rhizomes. So if you were at Susan's talk last night, you'll know that rhizomes are not roots, they're shoots. They're actually much more similarly related to these green branches here than they are to a root. And so they're stems that burrow underground really early in development. And what's important to note about them is that at some point they can re-emerge above ground, they green up, and then they can become reproductive. So these rhizomes are really a sort of separate axis of investment in vegetative biomass that has these reproductive consequences for the future. What was really interesting about this trait is it really only occurs in 3 species. And if you'll note, these are the 3 species that occur very largely at mid to high elevation habitats. So we also constructed a maximum likelihood phylogeny based on 60 whole genomes from across this group. And what we can see is that the occurrence of this trait happens multiple times across the phylogeny. Now, that's not to say that these are truly independent. We don't, there's multiple scenarios. We could have trait loss, we could have an ancestral polymorphism or introgression happening, but we know for sure that the trait itself is not monophyletic. Okay. And lastly, I'll just say that the sort of coincidence of this trait and high elevation habitats is really interesting because anyone who's been to a high elevation site for some period of time will know that high elevation sites often suffer really extreme weather events at bizarre times of the year, for example, like this hailstorm in early August. And so if you have this sort of set of biomass underground that's protected, you can imagine that this might be very useful for fitness in the long term for plants to be able to regenerate if some sort of extreme weather event happens. Okay, so just to wrap up this first half, do perennials vary in niche? The answer is yes. And there seems to be these differences in life history that are at least associated with this difference in elevation. So what's the genetic architecture? So to do this, I created an F2 mapping population between Mimulus and a coastal perennial Cotatus. These plants vary in a number of traits, including this investment in vegetative biomass, but also the production of these rhizomes. And so here is the total map. This is a lot going on, so I'm going to just focus on two aspects of it. The first is that you'll note that there are these two QTL regions, which I've shown in a red box. And these regions house these three different QTLs that affect flowering time, how many stolons or how many stems a plant made, and the highest node at which that stem arose. And so if you think about plant development, these are perhaps unsurprising traits to be co-localizing on a QTL map. And that's because essentially these are all the same trait. This is a developmental decision. So at each node on a plant has to make a decision as to whether to use their auxiliary meristems to make a flower bud or to make a stolon. And so it's perhaps unsurprising that these traits are so correlated, but what was really weird about this particular map is that if you dissect these two QTLs a little bit further, what you'll notice that their effects are opposite. So at the linkage group 4 QTL, it's the Cotatus allele that confers later reproduction and more stolons, and the opposite is true for the linkage group 13 QTL. And when you have this scenario in which you have these two relatively large effect QTL that have opposing directions, what happens is that in your F2 population, there is some large subset of F2s that have these hugely transgressive phenotypes. So these hybrids are really delayed at the onset of reproduction. They make a ton of stolons. They essentially just sort of sit around growing and bushing out. And we know from decades of field work that flowering time is one of the most strongly selected traits in nature. And so having these hybrids that have hugely delayed flowering might have some really severe fitness consequences. And I'm really excited to study that further. And the second aspect of the genetic architecture that I want to point out is the genetic ability to make these rhizomes that I've been going on about. So it's conferred by these two QTLs, which I'm showing in pink. And if we dissect these QTLs at the onset of reproductions, that middle developmental stage, what we see is that these QTLs have a hugely interactive effect. So it's really only individuals that are homozygous for both of these loci that are creating rhizomes at some appreciable rate. However, if we look at the effect of these QTLs across development, what we see is that this interaction effect, and in fact, the effect of this linkage group 9 QTL in general is completely ameliorated, such that it's only the linkage group 11 QTL that has an effect later in reproduction. And so one potential genetic model and what we think might be going on in the system is that the linkage group 11 QTL might influence whether or not a plant actually has the ability to make rhizomes, but the linkage group 9 QTL is influencing the expression of the linkage group 11 QTL. So it's influencing the timing of expression of this trait. And this bears out in our, if you just look at the phenotypes of the F2s through time. So really early in development, actually the ability to make rhizomes is largely recessive. So very few hybrids are making rhizomes at all. As development proceeds, that dominance coefficient increases. That's shown in this upper left-hand corner. And in fact, then by late development, what we see is that actually most hybrids are making rhizomes. So not only do these hybrids have this sort of transgressive phenotypes in the context of these life history strategies of investment, it also seems like they have some transgressive phenotypes in the context of timing of expression of these potentially ecologically important traits. And these also might have some fitness consequences in the wild. For example, hybrids might be able to make rhizomes, but if they make them super late in development, they might be competitively excluded below ground. Alternatively, if there's some really extreme weather event that kills off the plants and the hybrids haven't had time to make rhizomes before that happens, then of course they're going to have lower fitness than, for example, this high elevation species in a high elevation environment. Okay, so just to sum up, what I've shown you is that perennials of the Cotatus species complex vary both in ecology and in life history, and these seem to be associated. High elevation perennials exhibit this novel and very interesting trait of the production of rhizomes. And the genetic architecture of life history divergence is generally simple. However, there's some really unique quirks about it that make trait expression in hybrids potentially quite odd, with potentially some very severe fitness effects in nature. So with that, I'll just wrap up by saying thank you all so much for coming to my talk. Super happy to take questions. And I want to just especially thank my co-authors, John Willis, my PhD advisor, and particularly Maya Wilson-Brown, who's an undergrad in the lab who helped with this work a lot. Thank you so much.
Cool. Thank you very much. We have time for questions.
I haven't had anything come in yet. Oh, yes. Okay. Great talk. This is from Godspew. I can't read it. Great talk. If I have M. guttatus close to me, can I just pull them up to spot the rhizomes?
Yeah, probably. I mean, if you just kind of like, the rhizomes in these plants are very bizarre. So like in a grass, they're never above ground. But in these particular groups, they actually start above ground and they have this like really weird morphology where they just dive underground. So you might even be able to see those from a plant. Guttatus really doesn't make these rhizomes. There were two populations in which like one individual each made a single rhizome. Those were high elevation populations of Guttatus. But on average, especially along the coast, they do not make this trait. And they don't make it in a common garden either. However, if you live near the Cascades or near the Sierras, you might find Mimulus coriolinus or Mimulus decorus, which are phenotypically very, very similar to Guttatus. And they would also have rhizomes. So take a peek, dig them up.
Meredith says, awesome talk. Do you see any hybrids that are extreme in the other direction, i.e. that flower early?
Yeah, great question. I think the answer is no. And I think what's happening is that there is just some developmental constraint. So these are perennials. They're never going to start flowering at the first or second node. That's a very annualized trait. And so, yeah, we don't see transgressive phenotypes in the other direction. But I think that's a constraint of development.
Okay, Carol says, very interesting system and study, great talk, clear presentation of complicated results. Do you think flowering time influences pollination success?
What a great question. I have no idea. I have never studied pollination in this system. You know, there's a potential that, you know, the density of pollinators probably changes throughout the growing season of plants. And so later flowering plants might have a different pollinator community or a different density of pollinators. But yeah, we have no idea.
Cool. Thank you very much. Yeah, great talk. I think we need to move on to our next presenter. Our second to last presenter is Chuliang Song. The title is Untangling the Complexity of Priority Effects in Multi-Species Communities. Take it away. Chuliang, I can't hear you if you're talking.
Oh, okay. Sorry. Hi, everyone. Thanks so much to come to the talk. I'm Chuliang, currently a postdoc at McGill and U Toronto. Today, I'm very excited to present my work with Ted and Sergey on a new approach to untangle the complexity of priority effects in multi-species communities. We are all astonished by the amazing biodiversity in nature, from these tiny microbiomes in our gut to the rich insects in the forests. Almost all ecological communities that we see in nature are not shaped by all species coming in together simultaneously, but by some assembly processes. And these assembly processes will structure the very properties of ecological communities. Here we take a look at the hypothetical microbiome communities and see how the assembly history, the order of species arrivals, would impact the species compositions, which species are present in the community. So for example, starting from a green microbiome invading to the community, and it establishes, and then a yellow microbiome enters, and they both coexist. And then a red microbiome enters, and it would exclude the original green microbiome. And that green microbiome enters again, and then it may exclude the yellow microbiome. So you get a very dynamical changing species compositions depending sensitively on the very order of species assemblies. So here I've shown that assembly history matters to the species compositions. However, a fundamental challenge in community ecology is that it is often hard or even impossible to know which assembly history would take place in a given community. It is usually shaped by many processes, such as those large-scale spatial processes, which makes it impossible to know. So, for example, considering this microbiome community again, we can have infinitely many combinations of which species arrive first or later. And then this begets a question that with this inherent uncertainty about this assembly history, how much can we still tell about the species compositions in a community? So this leads us to the question about the predictability of species compositions under these intrinsically uncertain assembly histories. So suppose for the 3 species microbiome system, and we consider these 4 possible assembly histories. And then we see how this assembly history would impact this community. So suppose in the hypothetical ecological systems 1, in the end, we would have all 3 species coexist. So in this case, the assembly history does not seem to matter about the final species compositions. And then in another case, in another hypothetical ecological system 2, we see that the assembly history would lead to 2 possibilities of the final species compositions. And then let's consider yet another hypothetical ecological system where we would see 4 different final species compositions. And the priority factor refers to when the assembly history matters for the final species compositions. So for example, in this ecological systems 1, there's no priority fact. But for ecological system 2 and 3, they would exhibit priority fact. However, knowing only that they would exhibit priority fact or not are not enough, because while all non-priority facts are similar, the priority facts are unique one by one. So for example, in this ecological system 2 and ecological system 3, we can easily see that ecological system 2 is more predictable since it only has 2 outcomes, while ecological system 3 is less predictable because basically every different assembly history would lead to another species compositions. So this begets to the big question about what makes an ecological community more or less predictable under this intrinsically uncertain assembly history. This question is such a fundamental question in community ecology, and many wonderful work has been done. And most of them would fall into the category which I call the parametric approach. So this parametric approach would assume a form of population dynamics and usually a fixed structure of species interactions. So basically how strongly each species affect one another is fixed regardless of the assembly orders. And the classic result we know from the modern coexistence theory is that for 2 species, we would have these 3 different possibilities of ecological assembly. So for example, for the deterministic exclusion, regardless of the assembly order, you would end up with all 2 species coexisting. And then for the deterministic exclusion, regardless of who arrives first, the same species is going to exclude the other species. And then another case is called the priority effect, where the first arriving species would always exclude the late arriving species. So this basically is the main approach that people are taking to understand the priority effect in community ecology. However, a fundamental problem about this approach is that the very assumption of a fixed structure of species interactions is usually not empirically supported. Instead, ecologists have found that the structure of species interactions are very sensitive to assembly histories. For example, in this beautiful illustration drawn by Ted in his 2015 annual review paper, he has shown, for example, the very order of species arrivals would change the niches in the locations. So in other words, the assembly history itself changes the very niche relationships in the community. In other words, different assembly histories may lead to different structures of species interactions. Thus, by studying the structure of species, fix the structure of species interactions, we would limit the possibilities of what are empirically possible about priority effects. And thus, to overcome this problem, we use a concept called the assembly graph to address this problem. So the assembly graph basically try to compactly embed these effects of the order of species arrivals. So let's see an illustrative example. So suppose we have this species pool with 3 species. We named A, B, and C. And then we look at the coexistence combinations in the community. So for example, species A can persist in isolation. Species C can persist in isolation. Species B and C can coexist together. And all species together can also coexist. But all other species combinations cannot coexist in the community. And then the second thing we look at is the assembly pattern. So basically, for each coexistence combination, we need to we introduce the species that are absent in the community and to see the effect of the invasion. So for example, for the coexistence combination that only has species A, we introduce species B, C and B respectively into the community. So for example, here, by introducing species 3 into it, species C replaces species A. But when the species B invades, B cannot invade successfully. And so we can do it for all the different coexistent combination. And then we use the assembly graph to summarize all this information. So in the assembly graph, the nodes represent different species compositions. So for example, for 3 species, this corresponds to the communities that only has the species A, so it's B and C. And these ones correspond to the communities that has 2 species present. And this one corresponds to the case when all species are present in the community. And then we basically keep only the coexistent combinations. And then the edges correspond to how the species composition changes after the invasion of another species. So for example, focusing on the community that only has species A, when species C invades this community, we see that C replaces. So here from this community A, when C invades, it goes to C. And when B invades community A, B cannot invade successfully, and species A would still stay there. So in this case, we get self-loop. And similarly, we can do it for all other coexistent nodes. So for example, C, we have these 2 links, and BC, we have this 1 link. So basically, this assembly graph would effectively summarize all the information about the effect of the order of species introductions to the species compositions. And importantly, this non-parametric assembly graph, we do not need to assume how the structure of species interactions are going to look like, or how dependent it's going to be to the specific niche relationships. We simply look at all the possibilities of what are the possible assembly patterns. So for example, here we have presented the traditional parametric approach, where in these 3, we only have these 3 qualitative different assembly patterns. It actually can be proved if you're using a 2-species local Volterra dynamics with a fixed structure of species interactions. These are the only 3 possibilities that you can get for ecological assemblies. However, these are far from the full possibilities of the assembly patterns, even for 2 species. Using the non-parametric approach, we can show all the topologically different ecological assembly patterns. So for example, for 2 species, we actually have 6 different possibilities of ecological assemblies. And indeed, from an empirical assembly graph from this excellent paper in ecology from 2003, we can see some of these patterns are indeed present in the empirical assembly graphs. So in other words, this parametric approach indeed failed to incorporate the full possibilities of what ecological assembly can be like in real life. And then another question we ask is, how does these possibilities, the full landscape of priority effect look like when we increase the number of species present in the community? So here we show how the diversity of priority effect scales with the number of species. So here on the x-axis is the community size, how many species total in the species pool. And on the y-axis, it shows the number of topologically unique assembly graphs. So we have shown that for 2 species, we would have 6 different topological unique assembly graphs. And surprisingly, for merely 3 species, you get almost more than 40,000 possibilities of assembly graphs. So basically, taking a parametric approach is going to be almost impossible to grasp such a vast possibilities of what ecological assembly or priority effect can look like. And we can see that for merely 6 species, the possibility is far beyond the total number of all atoms in the universe. So indeed, the priority effect can take so many shapes that we are really underestimating previously about the full possibilities. However, these vast possibilities also makes us scared. How can we indeed understand anything about this priority effect? It seems impossible that we are handled with such a vast possibilities. So here we borrow the idea from those motifs in complex networks. We introduce these 4 dynamical sources, those topological features in assembly graph that can help us to break down a complex priority effect into pieces, into basic pieces. So here we introduce the first 3 dynamical sources introduced by Ted Fukami in his annual review paper. So the first one is called alternative stable states. So basically in the community, how many spatial compositions you can have that once you enter there, you can never leave it. Another one is called alternative transient passes. So basically going to the same stable state, how many different passes you can take to get there. And third one is called the composition cycles. So instead of ending some ending stable states, you constantly get some cycles in the species compositions. So let's take a look at how do these dynamical sources look like in an assembly graph. So here we present a hypothetical 3 species assembly graph. So here this has 3 species. So first we look at the alternative stable states. So as I've said before, those stable states refer to the states once you enter, you cannot leave. So topologically, it means that there is only ingoing links, but no outgoing links. So here, for example, we have 2 alternative stable states, AC and C. So you can see for AC, there's only ingoing links, but no outgoing links. Same for the species composition that only has species C. And then the second one is alternative transient passes. It basically means that how many passes would you have to get to the stable states. So in this hypothetical case, both stable states would only have 1 transient pass. And third one is the composition cycles. So basically how the composition would cycle when you assemble. So here we have a composition cycle of length 3. So AB can go to BC, can go to B, can go to AB. So there's a loop there. However, we found that actually these 3 dynamical sources are not enough. So we can look at another example with identical, the first 3 dynamical sources, two stable states. Each stable state have only 1 alternative transient passes and the composition cycle of length 3. However, these two assembly graph, the predictability differs strongly. So one is highly predictable, one is not so much predictable. about the species compositions. So actually we found that the missing dynamical source is the interaction between the alternative stable states with the composition cycles. So here we can see that as you are in the composition cycles, there's an escape from the cycle to the stable states. So basically you are not trapped into these composition cycles once you enter it. There's a way for you to escape. However, here in this case, once you enter the composition cycle, there is simply no way for it to go to a stable states. Thus, now we have these 4 dynamical sources, and we show that indeed these 4 dynamical sources, these topological features in the assembly graph can help explain the predictability of priority effects. So here on the x-axis, we have the observed predictability. So basically, we generate a bunch of assembly histories, and we run direct simulations on the assembly graph and to show the entropy of the final species compositions. And on the y-axis,
there's 3 minutes left.
Oh, okay, sure. And on the y-axis, we have this classified predictability. So basically, from the topological feature itself, so basically from every assembly graph, we only generate these 4 topological features, and we use a machine learning algorithm to predict to classify the predictability from this topological feature itself. And here each point corresponds to the number of topological different assembly graphs. As you can see, we have a super high R-square. So it's really a perfect explanation of these 4 dynamical sources really fully captures the predictability of priority effects. And then we can disentangle to see what is the relative importance of each dynamical sources. So interesting, we show that stable states does not seem to have so much importance in explaining the predictability of priority effect, despite the previous emphasis on the importance of the stable states. Instead, we show the transient passes, length of cycles, and indeed the interaction between cycle and stable states play a much important role for us to understand the predictability of priority effects. So to wrap up, to untangle the complexity of priority effects, first is the formulation. We introduce a non-parametric graph-based formalism to study priority effects. And second, we show the full diversity of priority effect actually is a super exponential increase with the community sizes. And third, we show that priority effects can be classified by decomposing them into 4 basic dynamical sources: the number of alternative stable states, the number of alternative transient passes, the length of composition cycles, and the interaction between alternative stable states and composition cycles. In the end, I'd like to thank my 2 collaborators, my advisor at MIT, Sergey, and our excellent collaborator, Ted. And thank you so much for your attention.
Thanks for the great talk. I'm thinking that in the name of time, we should probably move on to our last talk. But folks, if you have questions for Zhang, please enter them into the chat. And I just want to repeat again, find this, that, where is it? That, again, Mike Whitlock made a general Zoom room for people to get together between this and the natural history trivia. And the link to that is in the eSlack channel for this session and in the general channel. Okay, so our last speaker is Miguel Gomez, and I'm trying to see if Miguel is here. I'm unclear on this. No, okay. So in this case, I'm gonna go ahead and start this. So the title of this talk is "When Ecology Fails: Sexual Selection as a Species Coexistence Mechanism." Let me share my screen. Where is my thing? Crap, where is it? Oh, there it is.
I'm going to talk about how sexual selection and other reproductive interactions can promote species coexistence in ecological similar species. So 2 species are locally coexisting if they can maintain a population in the ecological conditions they experience. And this can be given by two different effects. It's the equalizing effects. These are fitness differences that can, if a species has higher fitness than the other one, it can outcompete it and eventually exclude it from the population. So these equalizing effects can reduce these fitness differences between species. And there's the stabilizing effects that has been traditionally defined as ecological differences. And this is if a species limits its own population growth rate, then it limits the population growth rate of the other species. I'm going to argue that these don't necessarily need to be ecological differences, but I will go for that. And importantly, this causes negative frequency dependence. So the population growth rate of either species is increased when they are rare, when the species frequency is low, and it decreases when they become common. If there's this negative frequency dependence, then no species can outcompete the other one. Each species will be rescued when rare, and they can coexist through time. Importantly, I'm going to talk specifically about locally species coexistence. Regional species coexistence can be maintained by other mechanisms like migration and stuff. So as I was saying, this, the stabilizing effects have been definitely defined as ecological differences, or at least studied more commonly as ecological differences. For example, foraging competition, this is resource partitioning, that kind of things. Phenology, predator susceptibility, or parasite susceptibility. But an intrinsic characteristic of every species is that they have sexual selection and reproductive interactions that can limit its own population growth rate, more than they limit the population growth rate of the other species. So this is a very noisy figure, but I will explain it in steps, so don't worry about it. This is just to show that there's at least 4 groups by which reproductive interactions can limit population growth rate, or prevent or promote species coexistence. This can be interactions, intra and interspecific interactions, and they can be between or within the sexes. So again, all my examples are going to be focused on local species coexistence. And these are intraspecific interactions between the sexes. So to the example of sexual conflict. Sexual conflict is expected to be more intense when rare, and more intense when common, because there's more males around trying to mate with the females. And sexual conflict can have important fitness consequences for the females, and just as a consequence in population fitness. So fitness is expected to decrease when common, because sexual conflict is more intense, and increase when rare, discussing this negative frequency dependence that can promote species coexistence. And there can be also interspecific interactions between the sexes. This is when males and females of the opposite species, males try to mate with these females, or they actually mate with the females. And these are costly for the females in the same way as sexual conflict. And it can be even more costly because if they actually reproduce, they will have no benefits. There's no viable offspring. And they work in the opposite direction as conspecific mating attempts, or last sexual conflict. These ones, heterospecific matings, are expected to be more intense when rare, just because the opposite species is common. And the fitness costs are decreasing the fitness when they are rare, heterospecifics are common, and the fitness increases when common. So this causes this positive frequency dependence and actually prevents species coexistence. And the interactions can also be within the sexes. I put the example of male-male competition. This is intraspecific within the sexes. There can also be female-female competition, but this is more rare. At least in the context of the species coexistence, it hasn't been studied. So male-male competition is expected to be more intense and more common, more frequent. Species is common. than when they're rare, and the cost in fitness is gonna be higher when common. So fitness is gonna decrease when common and increase when rare. The link between male fitness and population fitness is not as direct, but if the costs are sufficiently strong, it can be translated into population fitness. So yeah, male-male competition can cause negative frequency dependence and species coexistence. And the opposite occurs in interspecific interactions within the sexes. So males of opposite species competing. This is actually kind of common. Males can be territorial and just exclude every other male from the population, all their territories. And they might not have evolved species discrimination, so they just compete with every male that is around. Heterospecific male competition is expected to be stronger when rare, just because heterospecifics are common, and the fitness cost increases when rare. So the fitness increases in common that have positive frequency dependence, leading to species coexistence. So in the same way as ecological differences, reproductive interactions promote species coexistence, or can promote species coexistence when they occur within the species, so they're intraspecific interactions, and prevent coexistence when they are between species, or interspecific reproductive interactions. And we went about to test if these reproductive interactions can promote or prevent coexistence. We used these two species of damselflies, Eschinoria elegans and Enalagma sciatherum. These are two phenotypically very similar species. They overwinter as larvae, and in the south of Sweden, where we did the study, they can only emerge as adults in a narrow window of time. It's just June and July. They emerge at the same time, and they're active at the same time. And they just live as adults for a matter of days, a very short period of time. And because the time is reduced, in the time they can be adults reduced, and the time they survive as adults is very limited. They are in a very strong pressure to reproduce. And the reproductive biology is very peculiar, and it's kind of interesting and very costly for the females. So they're non-territorial, and males chase the females. There's usually many males chasing every female, until one male managed to clasp the female by the prothorax, as is shown in this picture. Males have a hook-like structure in the tip of the tail called claspers. So they clasp the female by the prothorax. And then the female has to bend her abdomen to reach the male genitalia. And the males remove the sperm from previous copulations. Thus last male paternity precedents, and then they inseminate the females. So this full reproductive cycle is quite costly. Chasing is obviously costly when they're in the tandem position and in the mating wheel, as it's called when they are copulating. In the mating wheel, they're both more visible to predators, and they cannot escape as well. And it can also take a long time. So they live very few days as adults, and there's just less foraging time and ovipositing time, the more they spend in this stage of reproduction. So we use these two species, and they're also very easy to study in the field and in the lab. So it was like an ideal system for us. And what we did with them, we did two different types of experiments. First, the behavioral mating trials, which we ask if there is frequency dependence in the reproductive interactions within and between species and within and between sexes. So the 4 groups that I was talking about. And then we did a mesocosm experiment to test if there was a fitness cost of these interactions. And this also answers, or it will also answer if there's species coexistence or exclusion. So these interactions and the cost in fitness prevent or promote coexistence. And then we did a series of field surveys to ask if what we found in our experiments actually can explain some of the demographic patterns that we see in nature. I'm going to start with the mating trials. This was done in a series of small cages. In each cage, we put males and females of both species in these two species frequencies. So when Ishinura was common and Enalagma was rare, and the opposite, when Enalagma was common and Ishinura was rare. And we mark each male that we put in each cage with this fluorescent color powder in the genital and in the claspers, in the tip of the tail, where the hook-like structure they use to clasp the females are here. And each male was painted with a different color. So we go by each individual. And after 24 hours in these cages, we recover the females, and by searching with a UV light, we could look for traces of the color powder in the female. So if the female have paint or traces of this paint in the neck, in the prothorax, then a male attempt to mate the female. If the female had a trace of color also in the genitals, then it was a mating. So then we could ID every male that attempt to mate or mate with the female. So with that, we could measure conspecific mating attempts, the number of conspecific mating attempts. This is a measure of, or a proxy of sexual conflict, and also heterospecific mating attempts. So these are intra and interspecific interactions between sexes. And we also measure male mating success. The idea of this is that if conspecific male-male competition is important, there will be less mating or reduced male mating success when they are common. If heterospecific male-male competition is important, there will be less or reduced male mating success when rare. So what do we found? These are results. In the x-axis is the frequency of the species, when rare and when common. The colors are the species. The red is Enalagma, Cepherum, and blue is Ishinura elegans. So in this figure in the left is conspecific clasping. It's a measure of sexual conflict. And conspecific claspings were more common, more frequent when the species were common. And heterospecific in both species, they follow the same pattern. And heterospecific claspings were exactly the opposite pattern. There were more heterospecific claspings when rare. When they are rare, heterospecifics are common. This was a lot stronger in Ishinura, and Enalagma didn't have that strong effect. And there was no effect of species frequency in male mating success. There was no difference in male mating success when common or when rare. So this was interesting because they follow exactly the opposite pattern, and we expect them to have the opposite pattern if they have an effect in fitness. So if conspecific claspings have a fitness cost, fitness is expected to be increased when rare than when common. If heterospecific claspings are more costly, then we'll expect them to have higher fitness when common than when rare. If they both have similar fitness cost or no cost at all, we will expect fitness to be similar when common and when rare. So to measure the fitness cost, we did a mesocosm experiment. We used these large outdoor cages. And we put males and females of both species in exactly the same frequencies as in the small mating trials. And here we mark every individual with a number in the wings. So every day we could go back, capture every individual in every cage to measure if they were alive or dead, and we had a measure of longevity with that. And we also let them have the full life cycle. So both species reproduce there. We have a water container with zooplankton and phytoplankton, and they oviposited there. The larvae develop there, and then they emerge as a new generation of adults. And we counted the number of adults that emerged in each cage. And with this, we could measure the female productivity. So the results from our mesocosm show negative frequency dependence. So this, again, the x-axis is the species frequency, very uncommon. And in the y-axis, we have the female productivity. This is the number of female offspring that emerge per every adult female that we put in a cage. And we also tested the total offspring instead of just the female offspring, and they have the same results. I'm just going to show this one. Again, the colors of the species. And yeah, there was higher fitness, higher female productivity when they were rare than when they were common. So with this figure, we can see that there's negative frequency dependence, species, this evidence that species are coexistent, and support the idea of sexual conflict being the mechanism behind this species coexistence. So finally, we wanted to ask if this actually happens in nature or it's just something else going on. And are we actually explaining some of the demographic patterns that we see in the field? So we did a population surveys, community surveys in 18 wild population, in 18 different localities in 2 continuous years, 2018 and 2019. We measure the species frequencies and the density. And our results are here in the left side. In the x-axis, we have the initial frequency. That's the frequency of each species in each locality in 2018. And in the y-axis, the colors are the species. In the y-axis, we have the density change. This is the density of each species in each locality in 2019 divided by the density in 2018. So 1 means that there's 1 individual in 2019 per every individual in 2018. So the population hasn't changed at all, it's stable. Above that increases, below that decreases. And we find the same pattern of negative frequency dependence that supports what we found in our experiments. So I'm just gonna conclude now. We found evidence that support sexual conflict being a mechanism that promote the species coexistence, at least of these 2 species of Härer and Alagma. One problem with our experiment is that we are not able to measure the effect of what's going on in the larvae. There could be larvae competition, but a previous study by Sipielski and colleagues found in 2 different Alagma species in North America that they are neutral. There's no effect species frequencies. And they have also found that this Härer, again, 2 different species of Härer and Alagma in North America, there's some differences in behavior during the larvae. Härer is more active, and it's also more susceptible to predators. We don't believe this is affecting our experiments just because we didn't have any predator in our mesocosms. But it will be interesting to in the future study what's the effect during the larvae and the adult to have the full life cycle and have a more thorough understanding of what's driving coexistence. But we have a very good evidence of a non-ecological mechanism. There's a intrinsic reproductive interactions that are promoting species coexistence in this Härer and Alagma species. So I'm just going to thank Adam Sipielski, Rachel Germain, Daisuke Kyogoku, Mark McPeak, and Eric Svensson. And a very special thanks to Sophie Lane and Ian Moody. They were undergrads when we did we worked doing this experiment together. They were awesome. They are both doing masters now. And when they decide to go for their PhD, you should definitely hire them.
So yeah, that was the end of it. Yep. So I want to thank all of the presenters here. In both the first and the second session for a very nice group of talks, and thanks everyone for being here. And chiming in with questions and whatnot. And I hope you can go hang out in the Zoom, and hope to see you at the, I should do my video. Hope to see you at the natural history trivia session that was been arranged by our grad student council for ASM. So thanks everyone. Good seeing you.